Good morning. It is a Monday. Let me know if you are here in the chat. So before we get started, I realize we're covering a lot of ground each day. Yesterday we went through the end of molecular genetics, or the biochemistry component of genetics, which is, it's more molecular genetics, but we went through the process of meiosis in great detail. And we just began our discussion of Mendelian inheritance. Any questions about any of that so far? Questions about how meiosis works? Before we get into today's actual content, I'm going to discuss what the project is for this unit. It is due Wednesday of this week at midnight, and I'd obviously like to give you as much time to work on it as possible. I meant to actually go over it on Friday, in case anybody wanted to start, because it is about meiosis, and now we've covered meiosis. Homozygous recessive alleles, so then guaranteed that all the children all get that trait. Yes, it is. Yep. That is, both parents only have the recessive trait. They can only give their kid the recessive trait. And if both parents have that, only recessives, then they can only give what they have. So they would have to give each child the recessive. We're going to do a bunch of examples of these together, but they would have to give both children that recessive trait, and then 100% of the children would be recessive in that case, which show their phenotype would be recessive. Phenotype is what you show. That's a good question, though. Where was I going with this? Right. So I'm going to go over the project right now. So if you guys want to look on the assignments tab, I'll pull it up on here as well. I don't know why, but my iPad has really struggled with this mirroring thing all of a sudden. It was working perfectly for forever, but all of a sudden it just decided that it doesn't want to do mirroring, really. I'm not 100% sure what's going on with that. Like, I can't even detect the computer here. Why is this not working? I may have to switch around how I do this. I wonder if I set my computer up here as a mobile hotspot and then connect to that. That's what I did at school. So, give me a second. I'm going to sort of rejig how this works here. It's an extra step that seems unnecessary, but it might be that the router's too far or some kind of interference. I don't know what's going on. Okay, now let's try it. I wonder if this will work better. I'm gonna try it this way. Oh, it is working now, which that's a good start. Okay. Assignments. Meiosis Live. So keep in mind, for this assignment, it is in fact live, which means that you need to present meiosis in some way. So for some reason, every quad, a number of students hand this in as just a slideshow or something like that. It can't just be a slideshow. It can be you presenting a slideshow, if that's how you choose to make meiosis live. But it has to be some type of live presentation. So let me show you what I'm looking for here. So in the past, students have done all kinds of things to demonstrate meiosis. Essentially, you are going to create a learning tool that is going to explain the process of meiosis to your fellow students. Okay, maybe someone who's never taken this course. Um, and there are some key things that you need to make sure that you discuss in your explanation of meiosis, and I'll go through them here. But it does need to be some type of live form. Essentially, you want to show where the chromatids are being copied, show them being copied, show the homologous pairs hugging each other as a tetrad, show the actual crossing over event taking place. It'd be a good idea to color code your chromatids, and then you can show exchanging of DNA. Very similar to the way I've done it in the note. You want to make sure that you're showing independent assortment, explaining it. So you do need to include all the important details. You need to show me the names of the. I'll go through this stuff in a second. Anyway, what you could do is a whole bunch of different things. Like I said, some people have done animations, so they've taken their phone and maybe done it with some pipe cleaners or done it with some pieces of plasticine or whatever you got home. It doesn't really matter. You could use colored pieces of paper. You could use white paper that you've colored a particular color. Whatever. I mean, it's it's pretty wide open in terms of what materials you want to use. Some people have done it as a computer animation. Not everybody will have those skills. So these are just some things that I've seen in the past that worked well. Some people did it as a flip book. The flip book is obviously technical and takes a long time to make a flip book. So, I mean, this is at your leisure, right? Like you can choose whatever method you feel fits your skill set the best. One student in the first quad did it as a news broadcast where they were like describing a news story about a cell going through meiosis, and then they just had like a little screen in the background that like showed what was happening and they were explaining it. Like, oh, that's kind of fun. The interview was another teacher suggested that to me as a potential way to do it. I'm not sure how you do the interview. The idea is that you're interviewing the cell, and the cell is like telling you about its process of meiosis, and so people have done this before as a like a collaboration. I should mention that. So we used to have we used to do this as a group project, but I don't do it as a group project anymore, and the reason being, in this environment, I have found that it's extremely difficult to keep students in contact with one another. And I had far too many problems with people bailing, people disappearing. Even if they were working with their good friends, some shady stuff went on for group work. And since I never see you, I'm going to say that that's going to be a no from me on the group work. So before you ask me if you can do it with somebody else, please don't do that. It has not worked out ever. Honestly, every single time I've done it, it has not worked out. So I don't do the group work thing anymore, which is it's a little bit too bad. But anyway, I feel like it's based on what's going on right now. I think not everybody is at their best, and there's nothing keeping people connected the way you would be if you like just saw that person every day and you're just like, hey, where's your thing? Whereas here you could just disappear, and then what do I do for that other person? I have no idea. So it was, anyway, we're not doing group work. You could do it as a whiteboard YouTube lecture. So there are a bunch of apps for this. There's actually some Chrome extensions that you can use to do this too. But the idea is that you have like a whiteboard in the background and you record your whiteboard, and then you like manipulate the stuff on the whiteboard while you're talking, and then you just do a screen record. There is another strategy that you could use where you could do the exact same thing but with a Google slideshow. You can draw objects that would be your chromatids, like just with like the drawing tool, and then you can manipulate them. You can click on them, slide them around while you're discussing it. Lots of people have done that, and that worked totally fine. So I mean, everybody has access to Google Slides, so you could do that. Or whatever, your idea. Now, I will say that if your idea is very unusual, maybe you want to run that by me. So I have had some ideas that did not work very well in the past, and I've had another group of students do the same idea and then have it work extremely well. So I'll give you a perfect example. I had some students that they wanted to do it as a dance. That's fun. So they did a dance, and then they explained like how their different like things that they were doing in the dance were like related to what was going on in meiosis. So this is like an extra layer. It's like meiosis as a metaphor. That's a real tricky thing. You have to be pretty on top of that in order to get that to work out, and actually explain what's going on in a way that makes sense, and make sure that you include all the relevant details of a meiosis. So I had one group that did that effectively, and it actually made sense at the end. And it was, I mean, they had textual descriptions at the bottom explaining what was going on in each part, and that was fine. And then I had another group do it, the exact same idea, but it was, I couldn't tell what was going on at all, and it wasn't explained properly, and it was terrible. So sorry, no offense to whoever did that, but it didn't meet the assignment expectations. So I'll leave it up to you, but let me know if you have an idea that you want to give it a shot. The idea here is that you're teaching this process of meiosis to somebody else. So what are you going to do? You have to pick an organism with a diploid number that is a 2n number. Remember, that's a full complement of chromosomes that is between 6 and 8 chromosomes. So that's the full chromosome number that it would be. Like in humans, it would be 46. Your organism is going to have between 6 and 8. And when I say select an organism, I mean actually pick a real species. So this is pretty easy to do. There's actually lots of resources for these on the internet, but you could just Google organisms or living things by chromosome number. There's like a Wikipedia article that lists like a couple hundred organisms and just by chromosome number. So like 1, 2, 3, 4, 5, 6 chromosomes. And then you just go through and find one that has between 6 and 8. So that's one resource. There's other resources for this to find them as well. But pick an even numbered one. We've been talking about meiosis as this idea that you go from 2n, the full complement, to 1n, half of your full complement in your gametes. And virtually all eukaryotes and sexually reproducing species do that. However, there are some exceptions in nature of species where the males have more chromosomes than the females, or the females have more than the males, or whatever. I mean, that does exist in nature. There are always exceptions to all these rules. Don't choose those. So if that's a species that you're coming across, don't pick something like that, because they have mechanisms in place in meiosis that account for those differences, which we obviously haven't discussed, and that's going to require a tremendous amount of extra work on your behalf to properly explain that. So don't do that. You're way overcomplicating this by choosing a species that's more difficult than it needs to be. So pick something between 2n, or a 2n between 6 and 8. 6 or 8, I should say, not 6 to 8, because 7 won't work. I really should change the wording there. But anyway, you can pick 6. So you don't have to pick 8. People are like, well, why would I pick 8? No reason. I just wanted to give you more options for organisms that you could choose. So, but it is more work, because it's just 2 additional chromosomes that you have to show going through the process. Because when you show the process, you're going to show exactly what it looks like in your target organism. Does your organism have 6 chromosomes? You have to show 6. That's the whole point. So far we've been doing our examples with only a few, like maybe a 2n number of 2, a very simple organism. Well, I want you to expand it a little bit in this explanation, and you're going to show me a 2n of 6 or 8. The other part of this is that you're going to describe it in your organism, so you need to tell me, is this a male that you're showing me, or is it a female? And where is it actually happening in your organism? Is it happening in the testicles of the muntjac deer? Is it happening in the ovaries of the muntjac deer? I'm just using as a random example, but that is a species. I believe they have 6 chromosomes. But anyway, you have to be specific. So you're showing me the process as it exists in that specific organism that you've chosen. Okay, and remember that male and female meiosis is a little bit different. Spermatogenesis and oogenesis are a little bit different. And so you do need to alter what you're showing me to reflect that. Okay, you need to make sure that you name every stage of meiosis 1 and 2. You need to make sure that you visualize every step. So you're showing me what is happening in every step and explaining what is happening in every step. Okay, this is very similar to the note. You need to make sure that you show the process of meiosis, or not meiosis, of crossing over. So you actually have to show the crossing over event happening. You need to describe it, you need to visualize it, and you need to explain why it's important. Okay, this is essential. Same thing for independent assortment. Remember, the independent assortment is when you select whether the chromosomes are going to go to the left or to the right with each homologous pair during metaphase 1. Okay, you need to show that. So you're going to show it, describe it, and tell me why it's significant. All right. And then lastly, oh, I already mentioned this. Yeah, you're going to make it specific to your organism, showing me where it's located, testicles, ovaries, which process are we looking at, spermatogenesis or oogenesis. So there's a fair bit of information there. I would go back to these instructions and double check that you've done all of those things before you decide to record, okay? I say that because for the most part, people's visualizations are great. They could do a good job. The spot where everybody loses marks is they just don't include all of these things. For example, they show crossing over, but they don't describe it. Or they show and describe it, but they don't tell me why it's important, or whatever. Okay, so that's the main place where people lose marks here. If you're looking for a completed one to use as an exemplar, you're like, hey, what does this look like when it's done and it like looks really good? A lovely student from a previous semester offered to let me use their assignment as an exemplar for you. So if you'd like to watch that movie, feel free to watch it. It is an excellent 4+ assignment. That's what it should look like. No, you don't necessarily have to include all the animations and everything. Like I said, there's various methods you can use, but this one includes all the required components and they're all well done. Okay, so I mentioned here just some things to think about while you're creating your project. The visualization component, that's the actual pictures themselves that you're showing, have to be created by you. So you can't just like find meiosis on the internet and we're not authorized. Kim, could you be a little bit more specific? It says you're not authorized where? Under what? Where would it say that? When what happens? Tell me a little bit more. And Julia, I'm going to get your question in 1 second. So you can't use pictures that you find on the internet? It says that? Okay, I will fix that. That doesn't make any sense, but I will fix that though. Sorry. So you have to create all the visuals. You actually have to show the correct number of chromosomes throughout the entire thing, as I already mentioned. You should make some attempt to show the movement that is occurring. What's going where and how are things moving in the various steps. It would be a really good idea to color coordinate. You don't have to color coordinate your chromosomes, but it makes it much more clear in terms of what's happening and what's going where. The video did work for you, student. Okay, interesting. It's uploaded. I'm gonna have to check. Give me 1 second. I can actually, Kim, I can actually impersonate you on Brightspace. Like pretend, like I can make my account pretend to be your account and click on it and see what happens. So let me just check to see if you're authorized. I don't really know what that means, but I'm gonna check. Anyway, the last thing I wanted to mention was I have some tips here for making a good Google Slides slideshow, and I'm gonna go through it real quick right now. So Jill, I'm also gonna get to your question. Oh, what's this? Oh. So this little Loom video that I have posted, tips for using Google Slides. I don't know why I called it that. It shows how you could potentially use Google Slides to make chromatids that you can like manipulate and move around in Google Slides. So I just made a little quick video of me making some so you can see how that process works if you're interested in using Google Slides. Okay, so what I mean, Jaleel, is that, for example, if I look up, I'm just going to look up the article right now, okay? So organism by chromosome number. Okay, there's a bunch of them. I'm going to use this Wikipedia article because it's just easy. It's an easy reference to use. Okay. The yellow fever mosquito has a 2N number of 6. Okay, that's its full chromosome number. So I could choose that as my species if I wanted, okay? Aedes aegypti. That means that when I actually do my visualization, oops, daisy. When I do my visualization, I actually have to show 6 chromosomes exist. Okay, 1, 2, 3, 4, 5. Oh, probably shouldn't have made that the same color. 5, 6. Okay, and then in interphase 1, you make an identical copy of each. Right? Those are going to be attached to each other at a centromere. And then there's going to be homologous pairs, right? So maybe this is the chromosome 1 maternal and chromosome 1 paternal. And chromosome 2 maternal, chromosome 2 paternal, chromosome 3 maternal, chromosome 3 paternal. Okay? That's what I mean, Jaleel, by a diploid number of 6. Okay, this organism has a total number of 6 chromosomes. When you finish and you have a haploid cell at the end, your haploid cell should have a half of 6 number, otherwise known as 3. Okay, so your finished cell will only have this in it, and you'll note that it's a random selection, right? That's how it works. That's the whole independent assortment thing. Now, these are not going to be complete chromosomes because they're going to have done a crossing over event with their homologous pair. So they won't be all one color any longer, which is something that you can show. Okay, hopefully I answered your question, Jaleel. Okay, cool. That's good, Ken. All right, then I won't investigate that then. Okay, so before we move on, I'm going to take questions from what we were working on yesterday. Does anybody have any questions about what the assignment entails? Tomorrow, the second block of the day is completely reserved for you to work on this. So that's approximately 2 hours. What we're doing in the first block is actually fairly short, assuming that we get everything done today that we need to get done. It's quite short. It's only maybe like a 30-minute activity. So you probably have a bunch of the first block as well. I have a feeling some of the stuff from today is going to get bumped a touch. So we may be doing the finishing off of this heredity stuff tomorrow. Anyone questions from yesterday's stuff? Yesterday's content, meiosis? We're going to do a quick meiosis review now to make sure everybody's good. So if you don't have a question, what you need right now in front of you is 2 colored pieces of paper. They don't have to be paper. They can be anything you like. A whiteboard will work with some markers. If you have string, you need 2 different colors of string, thread, yarn, pipe cleaners, pretty much anything you like. But you need 2 different colors of that thing, and you need to be able to cut the thing and reattach the thing. It works with strings and pipe cleaners and things like that. You can just reattach them. If you need tape to reattach them, that's totally fine. If you're using pieces of paper, you can reattach them with tape. But those are the materials that you're going to need in front of you. So if you don't have that, go grab that. I'm going to do it with pipe cleaners. Give me 2 seconds. I'm going to grab my pipe cleaners, and then we're going to get started here. I'll be right back. You go do the same. Okay, so sorry about that. I've got this. I'm gonna set something up here so that you're able to see what I'm doing on the desktop here. So. Got this from the school. It's like a stand that I can put here and put my iPad on it. So what I'm going to get you guys to do is try and create the same thing that I'm creating on your desk, ideally before I create it. So the idea is I'm going to ask you to show me what it looks like, or you're going to try and show what it looks like on your desk with prophase, metaphase, prophase 1, metaphase 2, prophase 1, metaphase 1, anaphase 1, telophase 1, and then the same thing with 2. I'm going to go through the process together with you, and then I'm going to get you to try and just show me the process. Okay, let's figure this out here. There, that works. So first of all, we're going to do this with an organism that has a 2n number of 2. Hi, Trenton. Sorry, what does in the assignment mean? Is that a question? This is what I'm doing here is similar to what you are going to do in the assignment. It's similar. I mean, in the assignment, you're going to be including headings for all of these, like you're going to be labeling with text each of the steps. You're going to make sure that you show the crossing over process, etc. So there'll be a little bit more detail than this. But this is the basic process that you're going to show me. I'm going to do it with an organism right now that is a 2n number of 2. And you're going to do it with an organism that's 2n number of 6 or 2n number of 8. If you're looking, Trenton, for an example of what the assignment should look like, I uploaded a exemplar of a completed version of Meiosis Live, so you can take a look at what it should look like when it's done. So I encourage you to do that. Please do that. It'll be very helpful for you. Okay, so I'm going to, like I said, do this with an organism with 2n equals 2. And I'm just going to use colored paper for my chromosomes. I thought I had pipe cleaners, but I think they may have gotten used up in a craft or something like that. That's okay. I can improvise. We'll just use paper. So keep in mind that these are the materials that I'm going to need here. I just have two things that are one color and 2 things that are the other color, and this can be anything for you. They can be pieces of plasticine or whatever you like. Okay. Okay. So let's imagine that my organism has a 2n number of 2. Okay. I'm not going to draw the nucleus or anything like that. I'll leave that up to your imagination. I'm just going to focus on what's going on with the DNA. Okay. So this DNA will be in the nucleus, and it would be in chromatin form, which means that it's just kind of laying out like an unwound noodle, just laying about inside of the nucleus. Okay, this is during interphase 1, before we're really getting into meiosis. Okay, so the key thing that happens for meiosis in interphase 1, this is during S phase, which is synthesis, which is a replication of all the DNA. So what we're going to end up with is a copy of each piece of DNA. I know this one's a little bit shorter. They should be exactly the same, but a copy of each piece of DNA. Okay, I'm using colors to denote which piece is which. Okay, and keep in mind that if this is a eukaryotic sexually reproducing organism, these have to be homologs, which means that they contain the same genetic information, but they're not identical to each other. So they have different versions of the genes. Those are called alleles. Alleles are versions. So for example, this one might have brown hair color, and this one might have blonde hair color or something like that. Okay, oopsie daisy. So I can even write that stuff right on here. You don't need to write these on, but just for illustrative purposes, I'm going to write it on. So let's say let's go right back here again to the beginning of prophase or beginning of interphase. Jeez, I gotta be careful with my my language use here. The beginning of interphase 1. Okay, and let's say that this one does have hair color on it. Okay, capital H, the dominant hair color. Let's say that's brown, and then this one has the recessive hair color gene on it, or recessive hair color allele, the version that is blonde. Okay, and then maybe there is a gene for tongue rolling. Well, we talked about that one yesterday, or on Friday rather. So let's say this one has the dominant can roll tongue, and then this one has the recessive cannot. Okay, obviously there's going to be more than 2 genes on a particular chromosome, but just obviously for demonstrative purposes. Okay, you don't have to draw these on yours. If you're using a piece of string or something, don't even worry about it. Okay, so we've got our total complete genome for our organism. Then in interphase 1, we're going to make an exact copy of each of these. Okay, now just from we're using colors, so it makes it obvious that there's an exact copy. By the way, follow along with whatever you've got at home. If you've got plasticine or pipe cleaners or whatever, follow along. Okay, we have an exact copy here of each, and I'm going to denote that by making exact copies of the alleles on it. Okay, you don't have to do this. The color should be enough to indicate that they're exactly the same. That's the purpose of the color, but I'm adding in the letter combinations because we're going to talk about the idea of alleles today in more detail, and this is sort of helpful for what we're going to discuss. So this is interphase 1. We've made a copy. All right. Then during prophase 1, hopefully people have a vague sense of what is going to happen here. In prophase 1, the homologs are going to get together and give each other a hug. Okay, this DNA is going to condense into chromatids, so we're going to see it condense into like the actual X shape that you see under the microscope, and they're going to get together and they're going to hug. When they hug, they do a process called crossing over, and that means that there's going to be an exchange of genetic information between homologs. Okay, so the homologs are different colors here, and so what I'm going to do to show that is I'm actually going to remove one of the genes, and you should remove a piece of each of yours as well. And it's good, they're going to switch between homologs. So we're actually going to get a switching of genetic information within this tetrad, like this. I thought I had tape sitting here, but apparently I don't. Where's my tape? It's been boring. Be right back. Okay, and then obviously once they switch, they're going to recombine. Yesterday, I keep saying yesterday, on Friday, we talked about some ways in which this process can go wrong. If you get an unequal switching of pieces and stuff like that, that's mutation. But we're going to assume that this is a perfect switch happening here. Okay, so that's prophase 1. We've done crossing over. Then, in metaphase 1, we're going to get these lining up across a center line in the center of the cell. Okay, and it won't just be this homologous pair. There'll be other homologous pairs depending on the organism, and they'll all be sitting on the metaphase plate. And which side you get the maternal and the paternal on is random. That's independent assortment. So it could sit like this on the metaphase plate, or it could sit like this on the metaphase plate. So which way it sits is random. It's going to go this way or it's going to go this way. That's independent assortment. And we talked on Friday about how that increases the amount of genetic diversity, which direction it goes in. Okay, that's also the reason that we do independent, or that we do crossing over. Okay, hopefully everybody's still with me here. Next, during anaphase 1. Anaphase 1 is when these two are going to get separated. Okay, looks like I got some room on the other side here. So they're going to get separated into separate cells. Okay, now they're in different cells. And in telophase 1, these are going to get put into nuclei, and they're going to decondense and go back into their chromatin form. Okay, so that, and then cytokinesis is the actual finishing of this. The cells are going to finally split apart and become two separate cells. Okay, so we've got 2 separate cells, and now they have half of the full genetic complement that they're supposed to have. So we call this 1N, even though technically it's slightly more than 1N information because of the bonus content that some of them are going to have because of the crossing over event. But we still call them 1N at this point because they're largely not 2N. Okay, so that's why the first meiosis 1, is called reduction division, because we've reduced the total amount of genetic information in each cell. Okay, so then meiosis 2, the purpose of that is going to be to put only 1 copy of each into each cell. So that's meiosis 2. In other words, we're going to split up the sisters. The sisters are still together right now. So yeah, 3 to 5 minutes is good. Some people had 7. 7's okay. 7's a little on the long side, but 3 to 5 minutes is good. In 5 minutes, you can do a very thorough job of going through the process. I'm taking much longer here, but that's because I'm not set up properly here. But you can do it in 5 minutes. Most of the best ones are about 5 minutes long. Like I said, some people took up to 7. 7's no big deal. Please don't do 10. Once you start getting into 10, that's 300 minutes or 4-5 hours of watching videos for me, which is a really, really long time for you to explain this process. So I would say keep it around 5. 7 is going to be your max. All right. Anyway, we've got 2 separate cells here now with this genetic information. We're going to go through the process again. So in interphase, the most important thing for you to remember is that you don't make a copy of the DNA the second time through. Okay, normally that happens during interphase, but it does not happen in interphase 2. So interphase 2, basically nothing happens. Okay, we do get centrioles being produced and things like that, but nothing with the DNA. Then in prophase 2, we're going to get the DNA condensing into the X form again. It's going to look like this under the microscope in an X, and the nucleus is going to dissolve. In metaphase, we're going to get the spindle fibers now lining these up on a metaphase plate. Okay, in each of these cells, there'll be a metaphase plate, and if there's more than one homologous pair, then they would all be lined up on that metaphase plate with one sister on either side. Okay, that's metaphase 2. Then in anaphase 2, we're going to get these being pulled apart. Okay, and then in telophase 2, these are going to get put in their own nuclei, 4 separate nuclei. They're going to unwind and go back into their chromatin form. And then cytokinesis 2, you're actually going to get a chop occurring. And then we've got 4 separate cells. Now, if you look at these 4 separate cells, you'll notice that their genetic information is totally different in each cell. This cell is not the same as this cell, is not the same as this cell, is not the same as this cell. And we've only done 2n equals 2. basically the simplest eukaryotic organism that you can have. Okay, and you have a 2n number of 46. So you can imagine how many different possible combinations there are. And this is with only one crossing over event happening. In real life, there could be hundreds of them on a single homologous pair, a single tetrad. So many, many, many possible combinations are going to exist here every time you go through this process, which is, of course, the point. And they're all haploid. So our organism has a 2n number of 2, but now the 1n number, which is what is in each of these gametes, is going to be 1. So if I were to take the genetic information from this sperm, okay, and bring in the genetic information from an egg, well, now I would end up with another 2n organism, a full genetic complement for a potential zygote for a baby. Okay, so and if we look at this, we can also predict what the outcome is going to be for that baby. So this is a big H that this baby is going to receive. And I believe we said that was hair color brown, I think. So it's going to show the dominant trait here for the top trait here for hair color. For tongue rolling, this one has a dominant tongue rollers yes and a recessive tongue rollers or a no tongue roll. But since it has the dominant allele, it's going to show its phenotype is going to be tongue rolling because this person has the dominant gene for tongue rolling. Now, if this was a little t in the end, if this was the genotype for this individual, little t, well, they would show no tongue rolling. And that would be if this was the baby that was formed, like we took a gamete from each parent. Were you guys able to follow along with this at home? Hopefully. With your own materials. Try going through this process on your own later. I highly recommend it because you really have to have a good, solid understanding of what's actually happening with these chromatids before you attempt to create your own visualization and show me. Okay, so you're going to want to get this process straight. I would start it with a 2n is 2, the way we've just done here, and then give it a shot and see if you can do it with 2n equals 6 or 2n equals 8 for whatever organism that you're choosing. Okay, see if you can keep everything straight and explain what's happening at each step. That's before you videotape it or before you do whatever you're planning on doing to visualize this. You don't have to videotape it, but you are going to make a video of some kind. Okay, I'm hoping that made things a little bit more clear in terms of what's going on with this process. Hopefully it did. I realize that this meiotic process is the most complicated thing probably that you do in grade 11 biology. So you know what, on the bright side, if you're comfortable with this, you did it. This is probably the hardest thing that you do this quad in terms of understanding a complex process. So hooray! It's all downhill from here. But if you're struggling a little bit with that, you're still not 100% sure what's going on, I highly recommend going back and watching the meiosis video that I posted, which goes through, it's like an animated thing that goes through the process again and with the description. And if you're still not sure, you go back and you watch that and you're like, I'm still not 100% sure. I have questions about this. That's totally fine. Come and visit me during Learning Block 3 today, and let's go through the process together. I'm happy to go through this again with you. And we can stop at any point, and I can answer your questions if that's where you're stuck. But for now, does anybody have any questions about what they've just seen, or is that okay? Just let me know if you have questions. No comment. All right, I'm gonna put this thing away. Later on in the quad, I'm hoping I can do a fetal pig dissection like that, although my wife has told me that she would very strongly prefer if I did not do a fetal pig dissection in the house, which I kind of understand. It doesn't smell the best. But we'll see. I may ask if it's possible for me to go in and work from the school for one day to do the fetal pig dissection. And if that's not possible, I do have a video of me doing it from last quad that I can show. Ideally, you have some window into the pig. I may use the video from last quad if that's the safest thing to do. I know I've already mentioned this, but things aren't going particularly well right now, so I wouldn't be surprised if the school said that they were not interested in that. There are people working from the school, but it's kept to an absolute minimum. So, okay. Anyway, we've gone over the assignment. We've gone back over the process of meiosis. I'm going to leave meiosis alone now, but just be aware that a firm understanding of what's going on during that is obviously essential for the assignment. You're going to tell me about it. And a large part of the quiz is made up of questions about the process of meiosis. So it is one of the main topics of genetics in grade 11. So anyway, let's move on. So on your content tab, I believe we are on day 8. We're going to continue. You'll notice that it says you will need part 3 of the course notes. We've already started part 3 yesterday. So if you want to open that up, we're going to keep going. So we were surveying some of our own genetic traits yesterday. We started with tongue rolling, I believe. Yes, and the dominant, they used R's. The actual letter combinations that you use when describing a particular trait are pretty much random. I mean, you can use whatever letter combinations you want for dominant and recessive. They tend to be related to the trait that you're analyzing, but it's not strictly necessary. I mean, you could basically use any letter. However, one thing to keep in mind is that it's a good idea to use letters where the capital letter, the uppercase letter, and the lowercase letter do not look the same or similar. So I didn't come up with this list here. This list is from a resource that I found online of some Mendelian traits. Remember that Mendelian traits are traits where one gene is equal to one trait. So in other words, the trait is decided by just one single gene. And there are very few of those in humans. So I got a list from an internet resource of some human Mendelian traits. In humans, there are very few Mendelian traits, but they don't always use great letter combinations here. For example, don't use S. S is the worst one to use because when you're, whoa, what just happened? When you're using S, this and this and this look very similar, especially if you have kind of sloppy writing. It starts becoming very difficult to tell whether or not you're talking about a dominant and a recessive or a recessive and a recessive or whatever. So I highly recommend that you avoid using those letters when you are establishing your own letter combinations for traits, because in most of the time you can just get to pick what letters you want to use. Sometimes in the textbook the questions will say, oh, this trait is dominant, capital P, and this trait is recessive, small p, or whatever. And even the textbook does a bad job at sometimes picking letters where the uppercase and lowercase look the same. But just as a heads up, it's not a good idea to do that. I've seen a lot of students confuse themselves. So, okay, we did tongue rolling. Free earlobe. So what that means is that if you look at their ear, is there dangly? Is there like a little bit that is dangling, or is it attached at the bottom to your head? Okay, so that's a Mendelian trait in humans. So if the dominant trait is the one with the hanging lobe, okay, so not attached, and so that's they're representing that with a capital E. I don't have mine as attached, okay, so that means that I have free earlobes. Free earlobes are dominant, and so my genotype is capital E, the dominant allele, and then the other allele we don't know. I could be heterozygous, which means that they're not the same allele, I could have a small e, or I could have two capital E's, two dominant alleles. There's no way to know. I'm showing the dominant trait, so there's no way to know by just looking at me. So I'm going to write that. Keep in mind that you could also write this as this and this. Oh, whoops. This and this. Okay, so that means the same thing. So in the textbook, they prefer to show you this little line that indicates that the other one is a mystery or whatever. Both of those mean the exact same thing, so you can write it either way. Okay, everybody recorded what their genotype is. If you have an attached earlobe at the bottom, it means that you would write two little e's because you are showing the recessive trait, which means that both of your alleles are going to be small e. Okay, if you ever show the recessive, you have to have both small e. If you're not sure, by the way, about some of these traits, yeah, hopefully you're not home alone. You may be home alone, but you could just ask whoever's at home with you, hey, do I have attached earlobes? If you're not sure. I'm sure they can tell you. Okay, hand clasp. So without even thinking about it, clasp your hands together like this. Okay, look at your thumb. Which thumb is on top? Okay, for me, when I clasp my hands together, my left thumb is on top. And believe it or not, that is a Mendelian trait in humans. It has to do with hand dominance, and hand dominance is a Mendelian trait. So if the left thumb is on top, that's dominant. Okay, you're going to show that with a capital C. I have left thumb, so that means I have the dominant trait, so my genotype would look like this. And again, C's are terrible to use for these letter combinations. Whoever makes these, like, how have they not figured this out? But don't use letters that look the same, the capital and the small letter. But that would be mine. If you have your right thumb on top, you're going to have two small c's as your genotype. Okay, next. Hold out your thumb. When you hold it out and do a hitchhiker's thumb, like as far back as you can go, does your thumb, like my thumb, curve back? Like it actually goes past vertical and curves back even more. Okay, that's what's known as hitchhiker's thumb. Okay, and that's actually the recessive trait, to have extra curve from straight. Some people, when they go up, they can just go to straight. Okay, straight is the furthest back that it goes. And that is not as common. Sorry. Wait. Which one's dominant here? Straight thumb is dominant. Straight thumb is dominant. So that's the dominant trait. So I don't have the dominant trait. I have the recessive. So that these are S's. Again, not a great one to use, but because mine's bent, I have little s, little s. But if you have a straight thumb, that's capital S, small s. Okay, sorry. Cedar, you asked a question. Is it more likely to have the dominant trait? It actually depends on the trait. So dominant does not mean more common. Some of these, if they don't affect the survival or reproduction of a species, then usually the distribution of these traits is completely random. It's in the next generation, the amount of people that have a particular trait might go up or it might go down. That's something called genetic drift, which we're going to talk about in the evolution unit just for a little bit. But something to keep in mind is that these are, I don't think any of these are related to survival. So they're not affected by evolution. Evolution only affects traits that increase your survival or reproductive ability. So because these don't affect your survival or reproduction, they're just drifting. That means in the population, these traits can go up, these traits can go down, and it's pretty much random. So whether it's dominant or recessive in any given generation is basically random. Now, some of these are more common in the population, and we're going to go through, once we're done this, and look at how common your particular genotype, that's your combination of all these different alleles, is. Okay, so somebody did this survey. I actually think the survey is like 5 or 10 years old, but it doesn't change that much in the population over 10 years. But you can see how common your particular genotype is, but it has nothing to do with dominant or recessive or like how important the particular trait is, because none of these are important. Now, if one of these traits was a survivability trait, it made humans more likely to survive and reproduce, which none of them are here. But if they did, then what typically happens is that evolution, selection, natural selection, causes that trait to become more common over time. So in each successive generation, things that allow you to better survive and reproduce increase in the general population. I'll give you a great example of that. Something that you wouldn't think would necessarily affect our survivability or reproduction, but blue eyes. Blue eyes is really the lack of pigmentation in the eye. It means that your retina, not your retina, your iris does not have very much melanin in it, or maybe none, no melanin. And melanin in the iris is actually responsible for making your iris work well, because your iris is responsible for blocking out light in bright environments. So if you have dark colored eyes, you actually have better, more acute vision in bright sunlight than people with blue eyes. Not by a lot, but it is a measurable difference. If you have darker eyes, you have better sun vision, basically. And that is a survival trait. So it's very unlikely in early humans that blue eyes were very common. And in fact, any records we have historically show that blue eyes were relatively uncommon in the ancient world. However, as human society moved away from hunter-gatherers, we became less hunter-gatherer and more agrarian. That is, we stopped constantly hunting for our food and started growing it. Blue eyes became more common in the population. And so you might be wondering, well, why were blue eyes becoming more common? Because people found them attractive is the reason. So it made you more sexually reproducible. That's something called sexual selection, where the selective pressure is actually coming from your partner. In other words, the males or the females of the species find a particular thing more attractive in a partner, and therefore it becomes more common in the population. So there's a good chance that the rise of blue eye color was a result of sexual selection in humans, because it is not a survival trait. It makes it more difficult for you to see in bright light, which is running contrary to your survival, especially if you're a hunter-gatherer. However, if you're not a hunter-gatherer, that selective pressure doesn't exist anymore, and so in that case you might see a change in terms of the number of a particular allele. But none of these, it doesn't apply to anything here that we're talking about, because these are just neither here nor there. This is just can you roll your tongue? Can your thumb do this or whatever? Like this is not anything related to your survival or reproducibility. I don't think you're going to be better at finding a mating partner because you're able to do a hitchhiker thumb or whatever. This is so there's no sexual selection acting on these either. Anyway, question. What if we only have one thumb that does it? Never heard of that. Is that true? Cool. I have no idea. I would say that that's probably environmental. So you might be hyper flexible in a particular joint. It's possible that that's due to an injury to one of your ligaments. I actually don't have an answer for you here, Michaela. I don't know what you would call that. It's never come up before. You have different mobility in one of your finger joints. I'm going to leave that one up to you. I don't have a good answer for you, so I would say pick one or the other. Sorry, because there's probably a number of things that could lead to that particular difference, so I'm not 100% sure. Anyway, I have hitchhiker finger. I'm bent, so that would be little b, little b. Next, the widow's peak. So a widow's peak is when your hair comes together and forms a peak at the front. I have one. I have a little peak where my hair goes whoop, and it comes to a peak in the middle. It's not flat across the top. You may need a mirror to figure this out for yourself, but if you have the widow's peak, that's dominant, and they're using W's here to describe that. I have the dominant trait. I do have a widow's peak. So this would be my genotype. Your results will vary. So if you don't have a widow's peak and you have a flat hairline across the top, that you would be recessive, or a little w, little w. Next, dimples. So that a dimple is when you have like a little impression in your chin right here. This is chin dimpling. If you're not sure what I'm talking about here, I'll look it up real quick, because people always ask about this one. Chin dimple. There, this person's got one. There's a chin dimple. So I have a little chin dimple, a little indent in the bottom of my chin. Some people have it, some people don't. If you do have a chin dimple, oh, never non-dimple. Do I not have a dimple? I thought I did. I must have looked. I must have like looked in the mirror. I don't have a mirror now. I thought I did have a chin dimple. I did this last quad, so I must have looked. I must have looked in the mirror. Yeah, I'll take a selfie. Huh. I don't have one. Okay. Well, anyway, if you're not sure, take a selfie. I don't have a chin dimple. So I would be recessive here. I have a non-dimpled chin, so little d, little d. Again, your results may vary. Mid-digital hair. Okay, so have a look at your fingers. There are 3 bones in each finger. Okay, top one, middle one, and bottom one that joins your wrist, or your hand bones rather. Top, middle, and then proximal, the closest to your body. So you want to look at the middle one, the middle section. Flip your hand over, have a look on the back. Do any of your mid digits, so that's the middle section of each finger, do any of them have hair on them? That's called mid-digital hair. Strange that that is a Mendelian trait in humans. I definitely have mid-digital hair. I have it on all my fingers, but some people will only have it on a single finger. It still counts. Although I lose it every time I do a barbecue, but mid-digital hair, I do have it, and it is dominant. So I have at least one dominant. That'd be my genotype. And then the last one, this is not, do you have blue eyes? It's important to note here. This is, do you have dark or light eyes? So there is a single gene that codes for significant eye melanin. Okay, so this in combination with a couple other different genes is what determines your eye color. But this gene only determines whether or not you have significant eye melanin or low eye melanin. So if you have significant eye melanin, then you will have dark colored eyes. So dark colored eyes are brown, or it's basically all the shades of dark eye colors. Dark brown, some people have almost black, hazel. Those are all considered to be dark eye pigments. If you have green, gray, or blue eyes, those are considered to be light eyes. In other words, you don't have significant eye melanin. Okay. If you look at my eyes, even just on the video feed here, they're dark. They're dark really under any lighting conditions. So I definitely have dark eye pigment. So I have the dominant trait here. Again, keep in mind that this is not whether or not you have blue eyes. Blue eyes specifically is not Mendelian. It is a combination of different genetic traits. One of them is this. So you have to have a light eye pigment. That is, you don't have the dark eye pigment allele. But that you also have to have other alleles present on other genes for blue eyes. I think there's 3 different genes actually that contribute to eye color. So eye color is not a Mendelian trait, but whether it's light or dark is Mendelian. Oddly enough, my son, who is only 5 months old, lovely boy. Let me show you a picture. He's one of the cutest kids in the world. Oh, you can see his eyes really clearly there. He has blue eyes. Yeah. And that's strange because he's the only one in the family that has blue eyes, except my mom. My mom has blue eyes, but I don't have blue eyes, and my wife doesn't have blue eyes, and her parents don't have blue eyes. Only my mom. So that actually says something about my genome. Maybe it's a little bit hard to see here, but I am heterozygous for this because I have to have the light eye gene somewhere in order to have passed it on to him. Now, I realize that all babies are generally born with blue eyes. However, usually around 4 months old, they begin to develop darker pigmentation in their eyes as their eyes transition to darker colors. And there is no transition in his eyes at all. So he doesn't appear to be changing his eye colors as he ages. He appears to still have blue eyes. So yes, sorry, Kim, exactly. You just pointed it out. They are born without any pigmentation because their iris cells have not started producing melanin yet. However, he doesn't have transitional eyes. He just has blue eyes. His eyes are not changing colors as he ages. So he's about 5 months old, and there's no hint that he has started changing eye color. So it's likely that he will have these blue eyes, which is kind of cool. It's good that he's the only one. So it's very, very likely, and you probably are not able to determine this for yourself, but it's very likely that this is actually my genotype here. And also, that is my wife's genotype because, again, neither of us have blue eyes. However, we produced a child with blue eyes. So it's very likely that both of us have this. And then when we did meiosis, the allele that was passed on to our gametes was, in fact, little p, little p. Those are supposed to be little p's, by the way. Again, p's are confusing. But you are right, Kim. Children are born with almost always born with blue eyes because their eyes haven't started producing pigment. But they usually transition within, or at least begin transitioning around 4 months old to having darker eyes. Okay, what I'm going to get you to do now is figure out how common is your genotype, your combination of these alleles. So, like I said, through survey, we've been able to figure out how common each of these alleles is in the general population. And the ones on the far left here are the ones that are the most common. And then as you go to the right, each trait is less and less common to have the dominant, that is. And so it's going to change how common your particular phenotype is based on, for example, if you had all recessive here, that would be the least common, the way that these are arranged. Now keep in mind that something being recessive does not mean that it's less common in the population. However, this table is set up so that the ones that are being measured in this table, it's not all of them, but the ones being measured in this table are the most common. The dominant is the most common, but it doesn't have to be that way. So for me personally, I have little r, so I would start at the bottom of this table, okay? If you have big R, you're gonna start up here. That's tongue rollers, okay? Tongue rollers are very common in the population, whereas non-tongue roller is relatively uncommon. Okay, and then you're going to go through and select each one. So I'm big C, I'm recessive for S, dominant for E, dominant for P. So my particular genotype would be combination of genes, and then the resulting phenotype, the phenotype is what it looks like, is relatively uncommon. That puts me down here at 21. So the combination is relatively uncommon in the population to have my combination of alleles. If you have dominant of everything, of these particular traits, that is the most common phenotype in the population. So you share that with the highest number of people. If you have recessive of all of these traits, that is the most unlikely combination. I've never had a student that was recessive in all of them. That is a number 32. If you were a number 32, tell me. I'm very curious about that. I've never run across a student that was a number 32 that had the most uncommon combination of all these alleles. It's fairly uncommon. It would be a pretty intriguing roll of the dice if you ended up at 32. I have had a few students that were that had phenotype number 1 though, which was dominant for everything. Anybody number 32? Just curious. Just curious. I'll take that as a no. I'd be surprised. I've taught this course maybe 25 times. Never had a student that was 32. Cool. Okay, I'm gonna take that as a no then. Anybody have phenotype number 1, the most common? I would say I usually have 1 per class. So it's fairly common. No one. Okay. Oh, Kim, you think you might, eh? You can roll your tongue, you clasp left over right, you have hitchhiker's thumb, or sorry, you have straight thumbs, and you have attached earlobes and dark colored eyes. Is that right? If you do, you're a number 1. Cool. Okay, we got at least 1. You don't get a prize or anything, Lillian, but it is kind of cool. No, you're not. Okay. No worries. So Lillian, you're the one in this class. That's phenotype number 1. It's so common that I usually have 1 in every class. Like I said, in teaching this course 25 times, I've never had a 32. So that combination of alleles is very uncommon in the population. But I'm sure one day I'll get it. And then I think if that ever happens, I'm going to actually do the stats. I'm going to figure out how many students I've taught in the number of times I've taught the course, and so how common it was in my class to get a 32. I'm very curious because I've never run into it, and eventually when I do, I'm sure I will run into it one day. I'm curious to know what the actual percentage is of students that I've taught that have 32, but it's right now the percentage is 0. So okay. Does everybody sort of get the idea of this, though? Of dominant and recessive, how you decide if you have the dominant trait, how you decide if you have the recessive trait, and how you can use your genotype, that is your combination of alleles, to determine your phenotype. Everybody's sort of okay with that? No one is looking at this and saying, I don't understand blank? If so, before we continue, tell me about that. "32 mean both your parents have homozygous recessive alleles." Yes, they have them. They don't need to be showing them. It just means that they have those alleles in their genotypes because they each gave you a copy. So for example, if you're little r, both of your parents could technically be showing the tongue roller. Okay, so they could be this. This would mean that both of your parents are tongue rollers. But when they made their gametes, they made little r gametes because it chooses, right, which one you get. You're going to get one or the other. And so then they passed on little r, and the other parent passed on little r, and they got an offspring that was little r, little r. But they may not be non-rollers themselves. They may be rollers, but they have the gene. I mean, they must have it. If you got it as a child, they must possess that gene somewhere in their genome. One of their copies of the gene is the recessive allele. And yes, each parent has the recessive allele somewhere in their genotype for every single trait. Now, not all the traits, because remember that to do this table, it's not every single trait that we just analyzed. It's only 1, 2, 3, 4, 5 of the traits. We did significantly more than 5, but of these 5, yes. Okay, I'm going to keep going. Keep in mind that what we're going to be doing now for the rest of genetics is we're going to be talking about analysis of heredity. We're going to be looking at what a parent has in their genotype and what they could potentially produce as offspring. So, for example, given that one parent has O-type blood and the other parent has A positive or whatever, or A blood, what are the chances that their child will have O-type blood or something like that? So that's hereditary analysis or genetic analysis. There are people that do genetic analysis for a living. They're called genetic counselors, and you can measure this information. Obviously, you can take genotypes of people through genetic testing, and then you can search through those genotypes for, it's mostly illnesses that people look for, but you can look for illnesses and you can look for all kinds of traits. And then a genetic counselor can help you determine what your chances are of getting particular illnesses based on your genetic traits. Or if you and your partner have a genetic analysis done, they can help you decide what are the chances that your child will have a particular genetic illness. And that's important for some parents because you may have in your genetic makeup a high potential for your child to have a devastating genetic illness. For example, if in your family you have a particular genetic illness, you may want your child to know that. I'll give you a great example. BRCA1 and BRCA2, those genes are genes that code for a predisposition. Predisposition means a likelihood that you will have breast cancer. If you have the BRCA2 gene, I believe that your chances of getting breast cancer are like 90% or something like that, or 95%. It's extremely high. So people that have that gene, the BRCA2 allele, I should say, it's an allele of a particular gene, it's a virtual guarantee that you're going to get breast cancer over your life. And people that have that allele usually opt to remove their breasts preemptively before they get breast cancer to prevent breast cancer from happening. They have all of their mammary tissue removed. Perfect example, Angelina Jolie had her breasts removed because she has the BRCA2 gene. So yeah, people do that, absolutely. And there are other genetic illnesses like that where you can take particular preemptive action in order to prevent problems. There are problems with cholesterol and things like that, where you can determine whether you have a very high genetic predisposition to a particular illness, colon cancer. There's a lot of cancer ones, in which case you may need to have enhanced monitoring. Maybe you need to have a check to make sure that you aren't developing cancer far more often than the general population would. So anyway, there's lots of reasons that this information is important. So we're going to be talking mostly about just like frivolous stuff, like eye color or whatever. Like these things are not particularly important, but there are lots of genetic traits that are of extremely high importance to people. And so it's not like that there's no point to knowing this information regarding genetics. Like this is extremely important. And for other reasons as well, for research purposes and whatever, it's important to know this stuff. But there are very real-world consequences to understanding expression of genes. So what we had previously been discussing in this entire unit is the idea of genetics. So that's just the science of genes, okay, and genetic variation in living things. We were talking about molecular genetics previous to this, and that is how you express your genes, how you go from having a code of DNA, a particular code, to making a particular protein. We call that protein synthesis or transcription and translation. That's molecular genetics, looking at the molecules that are involved in actual gene expression. What we're going to be discussing now for the sort of the second half is heredity, which is how is genetic information passed on from you to your offspring or from a parent to offspring. Okay, and meiosis is also focused on that. I mean, that's all about how you are creating a gamete to pass your genetic information on from parent to offspring. So I'm not going to go through this terminology because we mentioned most of this stuff already. The idea of dominant and recessive. We talked about alleles, which are versions of genes. By the way, probably all these words should make it into your vocabulary list. We talked about a genotype, which is which specific alleles you have. And we talked about the phenotype, which is what you look like. That's the physical outcome of the genotype. We talked about homozygous, when both alleles are the same, and heterozygous, when both alleles are different from each other. And then the rest of these here we're going to be discussing now. Okay, this is just review. We've talked about all this. One word, which I think I mentioned yesterday, but I'm not 100% sure, so I'm going to point it out again now, is that when we're talking about a specific gene, where you find that gene on a particular chromosome is called the locus. It's like, that's like the spot. Okay? If you're talking about multiple places on a chromosome, those would be the loci. Okay, so locus is singular, one locus, two loci. Those are just places. Okay? So if you look on homologs, homologous pairs, if you look at the same locus, the same place, you will find the same gene. Same genes, but different, possibly different alleles, versions of genes. Okay, I realize that I'm saying these terms over and over again, but it's because they tend to get mixed up, and this is like the core terminology. All right. Anyway, here's some examples here. I've just listed, okay, here's a locus, a location on a particular gene, and here's a homologous pair. And if you look at the two genes, it's the same gene in that location, but there are two different alleles for that gene. So we got big B and little b. Two different alleles, but the gene is the same. It's the same gene, codes for the same thing, and they're at the same spot or locus on this chromosome. This is just terminology. Okay, and then I drew some other ones on there. All right. And I'm going to skip over this. We basically covered all this in our example. So this right here is, again, just terminology. If you don't already have this, well, I don't know. You could write it down again. I don't think we expressly wrote this down anywhere, but a gene is a section of DNA that codes for a specific protein. An allele is a particular version. It codes for one particular outcome. Your set of genes, or your set of alleles, more specifically, would be like, that's called your genotype. So if you had big G, or big G, little g, little g, that would be your genotype. And your phenotype is what that looks like, what the physical expression is of that. Homozygous means they're the same, and heterozygous means they're different. I'm just gonna stretch. Oh, too much sitting. Oh, our bodies weren't meant for this. 20 minutes, we get a nice stretch. Okay, don't forget, I post all these. So this is posted. Okay, I'm going to skip over this. And I'm going to jump down here to Mendel's observations. Okay, this stuff that's above it here that talks about allele contribution and stuff, we covered this already. So we already discussed this. So Mendel, Gregor Mendel, cool guy. He was a monk. He grew a lot of peas. And his claim to fame is that he grew purebreds of peas, which means that they were peas with traits that every single time you bred them, you got the same trait. So if you get that, if you get a pure breeding version of something, what that means is that they are homozygous, because you breed them again and again and again, and no matter what, you never get a different trait showing for a particular gene. You never get a different allele showing. So they must be homozygous, because eventually if you keep breeding something and they're heterozygous, they have the recessive gene or they have a different allele somewhere, usually recessive, you're going to get one where the two recessive come together and they show. But if that never happens, if you keep breeding the peas and breeding them, and you always get the same allele, same trait showing, they're probably a purebred. So Mendel did that with peas. And then he took those purebred peas and he crossed them. So he started doing combinations of pea plants that were purebreds. And through those experiments, he was able to figure out basically all the information that we currently know about genetics. I mean, not everything, but everything that we know about heritability. So the idea that inherited traits are controlled by a pair of genes, this is going back to the homologous pair idea, that was Mendel. So Mendel figured that out, that it's a pair of genes that contribute to every particular trait. And that these are that's why they're called Mendelian traits. One gene equals one trait. Obviously, in humans, not everything is determined by a single pair. There may be multiple pairs that are involved. There often are. But in pea plants, a large percentage of the traits of pea plants are actually Mendelian. So he was lucky in that he was pretty much only examining Mendelian traits, which are named after him. And so that made his research much easier. Okay, but he did discover that idea that a pair is always involved in gene expression. These, by the way, are called Mendel's laws, or sometimes they're called Mendel's observations. And Mendel's laws are sort of like the foundation of our understanding of genetics. So Mendel is sometimes called the father of genetics because he figured out sort of all the basics of how genetics works based on some really interesting, intriguing math that he did. And we're going to look at one of those experiments in a second here. But anyway, pair of genes controls everything, and each version is called an allele. He also figured out this idea of the principle of dominance, which we've also talked about, which is that a dominant allele will override a recessive allele. So if you have the dominant allele, you will always show the dominant trait, even if you have a recessive allele as well. That is to say that, for example, if you have a pointy or straight hairline, even if you have the heterozygote, you're always going to get pointy, which we called widow's peak. But if you have two recessive, then there's no dominant trait to override the recessive, and then you're going to show the recessive trait. So that's called the principle of dominance. This idea that there are dominant traits and there are recessive traits, and the dominant always will override the recessive if it's present. Okay? So, and then there's just a little definition of what dominant and recessive mean. Dominant always masks. And so you can only ever see the dominant when there's a dominant and a recessive. Now, I should say that type of relationship between alleles, where one is dominant and one is recessive, is very common, but it is not the only type of gene expression. So we're going to talk about, this is also known as, by the way, complete dominance. I'm going to write that in here. Oops. What's going on? This is... So complete dominance is one way that genes can be expressed. That is, there's a dominant and there's a recessive, and whenever there's a dominant, the dominant always shows. Not all traits have that relationship between their alleles, where one always shows over the other one. And we're going to talk in this course today about some other types of inheritance that are not complete dominance, where one will always override the other one. Okay, but this is sort of the basics. And all of the traits that Mendel examined were completely dominant, or exhibited complete dominance. So in Mendel's world, every trait operated like this, where one trait was always dominant over the other. But in actual fact, not every single trait operates like this in real life. And like I said, we'll talk about some other examples. Okay, and then the last part here is this idea of the law of segregation, which really means that as long as genes are on different chromosomes, your chances of inheriting one particular set of alleles or a different particular set of alleles is completely random. And what that means is, for example, okay, I'm going to show you an example of a Mendel experiment, okay? So I'm going to establish two different alleles here. Capital R, we're going to say, represents green pea pods. Pea pods are the pods that peas come in on a pea plant. Okay, this is an actual experiment, by the way, that Mendel did. And little r represents yellow pea pods. Some pea plants have yellow pea pods. Okay, this is not meant to be a tongue twister. So we have the dominant trait, which is green, and the recessive trait, which is yellow. If I were to take a parent that was green, okay, but a purebred green. Purebred green means that both of its alleles are green, okay? And then I were to take a purebred yellow. Yellow has to be purebred because it's recessive. So we know for sure that it's going to be two little r's here, okay? And I were to breed them together. If you want to find out what the chances are of getting different outcomes here, we use what is called a Punnett square, okay? Invented by Reginald C. Punnett. And the Punnett square looks like this. It's when you take, I'm going to start it right from the beginning here. Okay, it's just a matrix, a grid. Okay, and on the top of the grid, you're going to put one parent. And you know what I'm going to do? Just to make this even clearer, let me color code them here. Okay, so I'm going to say, we'll call this the yellow parent and the green parent. No, I picked yellow. Okay, so just to make it clear, okay? So I'm going to put one parent on top. And I'm going to put the other parent on the side. This is called a Punnett square. So what the matrix does is it shows all the different ways that you can combine these two parents together. Okay? So you can have a big R from this parent and a little r from the top parent. Or you can have a big R from this parent and the other thing from the other parent, which is little r, keeping in mind that these are the same because they're both homozygous, right? Or we can have a big R and a little r, or a big R and a little r. Those are all the possible combinations we can get from these two parents. And if you look at the actual result that you make, they're all the same. So in this particular circumstance, if you take a purebred green pod and a purebred yellow pod, and you combine them together, every single offspring that you get is going to be, hopefully you can tell what color it'll be, has a big R, so these are going to be green, right? So Mendel did this, and he realized that every single pea plant that you make from this combination is always green, every single time. And so again, when you get a result like that, that's so consistent in an experiment, then you start thinking about why that's happening. And so this is what got Mendel thinking. Okay, I get green. I take a purebred yellow and a purebred green, and I mix them, and I always get green. There's something going on there that I would get green every single time. Why do I never see a yellow one in the offspring? That's weird. So this got Mendel thinking. From this, we can, by the way, rate what are called the genotypic ratios and the phenotypic ratios. The genotypic ratio is your chances of getting each type of offspring genes. Okay, so here, the genotype, the only genotype that you can get is big R, little r. Okay, so the genotypic ratio then is 100% big R, little r. You're going to be defining this later when you do your problem solving. You're going to be telling me what the genotypic ratios are in your problems that you're going to do. And then you also look at this and you say, okay, well, what's the phenotypic ratio? Phenotype, remember, is the actual outcome. In this case, what color is it going to be? So they're all green pea pod in the end. And so the phenotypic ratio is 100% green. That's the ratio of different colors or outcomes. Okay, it's not always going to be as clear cut as this. Obviously, this is like the easiest type of one, but here you're going to get 100% green. Okay, so Mendel looked at this and said, that's weird. Why are they always green? So Mendel said, okay, I'm going to do another experiment here. I'm going to take these offspring from the first union here, the green and the yellow, and I'm going to breed them with each other. I'm going to take the babies and breed the babies. So by the way, what we have here, big R, little r, all these babies that we've produced, are what are known as the F1 generation, or the first filial generation. The word filial, a filly is a horse, a baby horse. And first filial means like the first generation of baby horses. And the reason why the horse terminology comes in here is that one of the original uses of this was for horse breeding. As you might be able to imagine, people were and still are breeding horses for particular traits. And so this was often used for horses. And so the first generation in a genetics experiment is known as the first filial generation. So that's the first generation that we produced. Big R, little r is the first filial generation. Okay, so if we take the first filial generation and now we cross them, this is what's known as an F1 cross. Okay, an F1 cross just means you're crossing the first filial generation. This, by the way, is called a P1 cross, the first one at the top, because P1 is like the parental cross, the original cross that you're doing. So this is a P1 cross. P1 is just the original parent generation that you have. Sometimes just called P cross. Okay, so we got the parent generation, and then we have an F1 cross, which is we're taking the first filial generation and we're crossing them. Okay, so we've got big R, little r times big R, little r. That's also, if you were to look at the phenotype of that, that would be green times green. Okay, we're going to mate a green with a green. We're going to do the exact same thing with our Punnett square. So let me erase all this here. Okay, so we have, oh, it's missing a line. We have our green offspring here and our green offspring, and we're going to mate them together. And now when we do this, we're going to get big R, big R. Okay, that's if both parents give a big R. Or we're going to get big R, little r. Or we're going to get big R, little r. Or if both parents give a little r, we're going to get little r, little r. Anybody have any questions about how these Punnett squares work? All we're doing is we're looking at the different ways that each parent could give their allele. So each parent can either give one or the other, and the chances of giving one or the other, of it being this one or this one, is completely random. So going back to meiosis, you'll remember that which allele you get at the end, whether it's the one from their maternal or paternal side, is completely random which one you get. Okay, and because it's completely random, the outcome that you're going to get at the end here for each of these four options is exactly the same. What I mean by exactly the same is that 25% of the time, let's see if I can write this really small, 25% of the time, you're going to get this, and 25% of the time, you're going to get this, and 25% of the time, you're going to get this, and 25% of the time, you're going to get this. Okay, so it's a 25% chance for each of these. It's a dice roll. It's a four-sided dice roll. Okay, if you can have a four-sided dice. I guess it'd be a triangular pyramid or something dice, but yeah, you could have a 4-sided dice. So you roll 4-sided dice. It's going to be one of these. Okay. By the way, going back, that's Mendel's third law. I meant to mention this when we did it the first time, but I forgot. That's called the law of segregation. So essentially, what that means is the law of segregation means that because you independently assorted stuff, remember independent assortment from meiosis, the alleles went one way or the other way, and it's random. Because it's random, the chances of getting any one particular allele in your offspring from the parent is a random chance, which means it's going to be an equal percentage for each of these options. If we didn't have independent assortment and it wasn't random, the chances of getting one of these combinations or the other combination would be different. In other words, it might be more likely to get this one or more likely to get this one or more likely to get this one. But in actual reality, because of independent assortment, it's completely random and you have the same chance of getting any of these. Okay, a 1 in 4 chance. So Mendel looks at this and says, okay, well, what are my genotypic ratios that I get? And again, you guys are going to be doing some analysis of genotypic ratios as well. So my genotypic ratios here are, well, first of all, let's write down the genotypes that we have. We've got big R, we've got big R, and we've got little r. Those are our genotypes that we've got. Why didn't that show up? Little r. Oh, there it goes. Oh, geez. This thing's on a crazy delay all of a sudden. Okay, I'm going to go back. I'm really, really hoping that this is going to save this properly. Saved? I'm going to be bummed if it didn't save it. And I'm just going to restart this. Sometimes it gets bogged down. And you got to restart it. All right. Yeah, obviously there's like a huge delay. Sorry. This is the one I want. Whoops. It sent me back up. Please tell me it didn't delete everything. No, it didn't. Okay. Genotypic ratios. So these are not an equal ratio to each other. So if we actually look at the ratio, there is 1 big R to 2 big R little r's to 1 little r. So those are the ratios of the different genotypes that you get one to the other. Another way of writing this, if you don't want to write it as a ratio, you can also write it as a percentage. So 1 out of 4 is big R. So that's 25% big R. 2 out of 4 is big R, little r. That's 50%, right? 50% big R, little r. And little r is 1 out of 4. So it's 25% little r. Okay, so those are the genotypic ratios, the ratios of one to the other. And what this means is that if you were to breed these two plants together, exactly 25% of them, well, I should say exactly. It's random. There's randomness here, right? But the more you breed, the more you're going to see this trend, okay? So it's going to be approximate because it's random, but approximately 25% of the plants that you get are big R, and 50% are going to be big R, little r, and 25% are going to be little r, okay? So if we look at phenotypic ratios then, remember phenotype is the outcome, what it physically looks like. And we go back here and look at what these will have in terms of phenotype. So big R, remember, codes for green. So this is going to be green. This has the dominant, it'll be green. This has the dominant, it'll be green. And this is homozygous recessive. Homozygous means both are the same. Homozygous recessive, which is going to be yellow. So the phenotypic ratios here are, so it's either green or yellow. And the ratio here is 3 green to 1 yellow. And another way to write that is, 75% green, 3 out of 4, and 25% yellow. So try to imagine being in Mendel's shoes here. Okay, and Mendel did this, by the way, with more than one single trait. He did, there's a whole bunch of different traits that you can breed pea plants for. They're all Mendelian. And so this experiment was repeated with a whole bunch of different traits. But imagine that Mendel does this, and every single time he does it, he gets 75% green and 25% yellow when he breeds the children of this first pair. Okay, so he gets this first pair, the first filial, then breeds the first filial, an F1 cross, and every single time he's taking all green plants and getting 75% green and 25% yellow. Again, when you see a result like that, you have to start thinking as a scientist, something is going on here that is creating this unusual situation. What is making this happen every single time I do this experiment? And so all that Mendel had to go by was just the phenotypes, right? Remember, he knew nothing about the genetics. He knew nothing about the fact that there are two traits. And just simply based on that, the fact that he always got this result, 75% green and 25% yellow, or whatever for whatever traits. Again, he did this with multiple traits. He was able to work backwards and determine, well, it must work like this, which is a pretty amazing logical leap, which is that he worked backwards and determined, well, there must be two copies for this trait on each plant, and it must have some kind of dominance and recessive quality to it in order to always produce these results every single time I do it. I mean, that's amazing. That's an amazing logic connection to work backwards from the results and then get the math, get the core process that results in this particular outcome. And it was a guess at the time. Okay, so he was working backwards and he said, okay, well, there must be a dominant and a recessive, and we must be getting genotypes that look like this. Again, this is all developed by Mendel. But then he used those results, his guess, his theory, and by the way, this is what a theory is in science, which is that it's your best guess to how things work based on the evidence, and all the evidence that he had suggested that this was in fact how it worked. Okay, and then he said, okay, well, let me design some other experiments, and if I'm right, these are the outcomes that I will get. So I'm gonna breed these, and if this is correct, I'll always get 25% this, I'll always get 75% this. And then he did those experiments, and he did get those results. Those results confirmed his theory that this must operate on a system of two alleles for every trait, and then there's a dominant and a recessive. And he did a whole bunch of additional experiments in pea plants, combining traits together and stuff like that, and looking at the outcome. And his predictions were always correct. It always suggested that this was in fact true. And then later on, when we were able to actually do the genetics testing, and this is like over 100 years later, we were able to do the genetics testing and actually look at the chromosomes and sequence the genome and look at the alleles. It's true. This person figured this out without having any biochemistry whatsoever. This person had never seen a chromosome under the microscope before, had literally never seen the genetic information of an organism, but was able to figure out how this inheritance pattern worked in living things. I mean, that's amazing. That is groundbreaking. And that's the scientific process. That's the real magic of the scientific process, which is that you're able to make some observations, make a guess which supports those observations, and then test it. And then you come up with often a process that is accurate. It accurately describes what is going on at the molecular level, which I think is really amazing. Anyway, that's Gregor Mendel. That's why he's the father of genetics. Don't forget about Mendel's 3 laws. Inherited traits are always controlled by a pair, and that is true for Mendelian traits. The principle of dominance means that a dominant gene is always dominant over a recessive gene. It always shows if it's there, if it's present. Keep in mind that principle of dominance is modified by some of the other things that we're going to talk about in this unit. In other words, complete dominance, as shown here, is not how every single trait is expressed. And then this last idea here of the law of segregation. which just means that it's random which combination you get. Of the 4 options, which one you roll on the dice is completely random. Okay, so that's where we got this information from, this idea. What we're going to do when we come back, because it is break time, is I'm going to go through some examples of actual crosses. We're going to do some problem solving using this technique of using Punnett squares. And we're going to talk about some other types of inheritance that are not complete dominance and incomplete dominance, or sorry, that are not complete dominance. Okay, I'm going to give you guys a break. Enjoy your break, and then I'm going to give you some homework questions to kind of sort of practice this stuff out. Let me know if you need anything, but I am going to probably eat a granola bar. Okay, so taking a quick look here at what we've covered. So what we've gone over here is sort of this basic idea of heredity, and how one would problem solve a little bit. We're going to go through that technique in a little bit more detail. We're going to repeat it and look at using Punnett squares to analyze inheritance and like the percentage of what potential outcome you would get. Those are the genotypic and phenotypic ratios. Sort of a standard set of things that you would always include in a genetics problem. Just be aware when we're done, I'm going to get you guys to be working on question 5 on page 193. There's some practice questions that go through the solutions with you. That's on page 191, numbers 1 and 2. You should try those. Those are practice genetics problem solving questions. We're going to do this after the lesson is over, and I'm just letting you know where we're headed here. And then I have 2 practice quizzes here. One of them is a simple quiz about the outcome of a basic genetics problem. One single trait, general inheritance, like just like the P problem that we did together a minute ago. And then quiz number 2 here is on using Punnett squares and the idea of the test cross. So the test cross is what we're about to talk about now, and we'll get to that in 1 second. Then what we're going to do together in the lesson is we're going to talk. Why isn't this working? There we go. We're going to talk about, as I mentioned, test crosses and incomplete and codominance. So incomplete and codominance are 2 other types of inheritance. The last part here, blood types and multiple allelic inheritance, I think that we're going to save that lesson for the beginning of tomorrow's class. I think if I try and squeeze that all in today, that'll be too much. Let's get through today. Let's get through this stuff, incomplete and complete dominance, or sorry, incomplete and codominance, and then I'm going to give you an opportunity to work on all these practice questions. And if we're nearing, the 12:30 mark and everybody's done, which I feel is going to be unlikely, but if it is, if things are going well at that point, then I'm going to just keep going with the next topic. But I think I may have overloaded it a touch again, so I may leave this lesson on blood typing till the beginning of tomorrow. So that actually is totally fine, because this lesson on blood types and multiple allelic inheritance is about an hour, and then the other activity that we're going to do, which is on genetic illnesses, is fairly short. It only takes about half an hour, 40 minutes. So we'll probably finish that during learning block 1 tomorrow, and then I still have the entire second learning block to give you to work on your projects. So that's I think that's how we're going to break down. I'll let you know, but I don't think we're going to get to this blood type stuff, which is this learning block 2, blood types and multiple alleles. I think we're going to bump this to tomorrow. Okay, so let me keep going. What we're going to do now is do some genetics problem solving. I'm going to walk you through some genetics problems. I'm going to erase this stuff here. I'm going to go through it with you right now. And these are the standard set of steps that you are always going to use when you are solving genetics problems. Okay, so here's a standard genetics problem that you might see. In humans, brown eyes are dominant over blue. typically I'm going to tell you that. Either I'm going to tell you which one is dominant, or the nature of the question is going to make it clear which is dominant and which is recessive. So for example, if it tells you that the heterozygote, that is when you have the dominant and the recessive, if it shows this trait, this phenotype, well, that's going to be the dominant trait in that case. So there'll be some indicator within the question which one is dominant and which one is recessive. In this question, it says, if a homozygous brown-eyed man is crossed with a blue-eyed woman, what are the expected genotypic and phenotypic ratios of the F1 generation? Remember, the F1 means first filial. It means of their children. Okay, the children of these particular parents, the first filial. You can also ask about the second filial generation, and the second filial generation is the children of the children. That would be the F2 generation. And I don't think I really get to that. Normally the unit is quite a bit longer when we run it in the standard course, and we actually get into second filial generation analysis and a couple of other more complex things that we never touch on in this course. It doesn't really matter. It's not super important for grade 11, but we're only ever going to talk about the first filial generation, if I remember correctly. So the first thing you need to do when you get a problem like this is you need to establish what are called let statements. So I have them numbered here. That's the number 1 in the corner here. That's the first step. So step 1 is establish what letters, what lettering you're going to use to symbolize your dominant and recessive. And remember, dominant is going to be a capital letter and recessive is going to be a small letter. You can use whatever letters you want. I highly recommend using letters where the upper and lowercase look different. I think I mentioned this on the quiz too, like what's wrong with this particular lettering for this genotype. That's the answer. The answer is that I'm pretty sure, if I remember correctly, it's that the upper and lowercase look similar. So just watch out for that. But I've chosen B's here. Okay, so I'm saying let B, capital B, be the allele for brown eyes, and let small b be the allele for blue eyes. Okay, so we got brown and blue, and brown is dominant. It's going to be the capital letter. Okay, so that's the nomenclature that you're going to use for, this is a simple dominant and recessive trait, which is also known as complete dominance. So that is the type of inheritance that is happening here. So complete dominance just means that there is a dominant gene and a recessive gene. Okay, it's very straightforward. So when you have complete dominance, you use a capital letter and a small letter. There are other types of inheritance, which we're going to talk about later. But for complete dominance, use capital and a small letter. Then you're going to tell me the cross, okay? So what that means is you're going to tell me the genotypes of the parents that are being crossed and the phenotypes of the parents. Now, in every problem, you're not necessarily going to know the genotype. Sometimes you're being asked to figure out the genotype of the parents. So what you write in here is what the parents could be. Okay, so in some cases, you're going to be saying something like big B blank or something like that if you don't know the full genotype of the parent. And maybe you will figure that out through answering the question. But in this case, we do know because we were told. So it says a homozygous brown-eyed man. Okay, so homozygous means that both alleles are the same, and brown is capital B. Okay, so homozygous brown-eyed man and a blue-eyed woman. Okay, there's only one genotype that can produce blue eyes, and that's little b, right? So those are the genotypes of the parents. Then you're going to tell me the phenotypes of the parents. And by the way, you don't have to do these two steps in this order. You can do the phenotypes first and then the genotypes, but you do need to indicate what they are when you're solving one of these problems. So we know the genotypes of the parents. Now the phenotypes of the parents are fairly straightforward here. We're told it in the question. This is brown and this is blue. Okay, any questions so far? I think this is fairly straightforward here. These are the steps that you should pretty much always include in a genetics problem-solving problem. This helps keep all of your information organized. In other areas of science or in math, you might use the GRASS method for problem solving. This is kind of like the GRASS method, but for genetics problem solving. It doesn't have a name or anything, but this is this is just like a standard breakdown for how you would do a genetics problem. Okay, all right. So we have let statements, genotype of parent, phenotype of parent. Step number 4 is that you're going to produce the Punnett square. Okay, so you're going to put the parent's genotype, one of the parents on the top, and one of the parents on the left. Now here, I've indicated which one is male and which one is female. It's not super important for this problem, okay? You can indicate that information, which is male and female, but you do not have to do that. Do you know the blue eyes are going to be both small b's? That's right, because the trait is recessive. So the only way that someone would have blue eyes is if they have both small b's. If they even had one big B, it would be brown. So, okay, and then you solve your Punnett square. So I'm going to solve this. So this is going to be big B, little b, big B, little b. I tend to write the dominant allele first every time, because if you do that, it'll help you keep everything straight. If you write some of them this way and then some of them this way and switch them around, at the end when you're doing your counts for the ratios, you might accidentally not put two things that are identical together, which is problematic. So I personally always write the dominant and then the recessive. The textbook does that too, and I highly recommend you to do it as well. But it's not like a rule. Nobody's going to look at that in university and be like, oh, that's not how you write that. But I highly recommend it. It will help you keep everything straight. And then we've got a big B, little b, and a big B, little b. So you'll notice that these are, yes, they are all the same. They all are big B, little b, and big B is for brown. And so the phenotype that we're going to get here is all brown. You don't actually have to indicate this on your Punnett square. I'm writing it on here to make it clearer. But you don't actually have to show which color, like if you don't have highlighters or whatever, no big deal. Okay, so these are the genotypes of their potential children, and you'll notice that they're all the same. So when we write the genotypic ratios, that's our next step, is to indicate, that's step 5, indicate the genotypic ratios. Well, we only have one genotype in this case, which is big B, little b. So we're going to say that. We have big B, little b, and it's 100%. For the phenotypic ratios, remember phenotype is, and that's step number 6, phenotype is the actual expression of the genes. So what does big B, little b look like? And in this case, it's brown eyes, and it's 100%. Try to put eyes here, brown eyes, 100%. Okay, the last component, or step 7, is, which I didn't write, I should have wrote it. Step 7 is the therefore statement that you put at the end. So you actually have to answer the question. So the question here is, what are the expected genotypic and phenotypic ratios of the F1 generation? Okay, so this is our F1 generation that we just found here. And so you just answer your question in words. I should probably make this way smaller to make it fit. Born. Okay, that's just answering the question. Okay, so that's a standard problem set, a standard solution for a genetics problem. Now, the quiz here is multiple choice. So it was very difficult for us to do any kind of paper testing. We started off doing that at the very beginning of the year, and it was just a nightmare due to I can't touch your papers, and you can't touch your papers. We had students take a picture of their test and send it in, and then half of them you couldn't read properly, and they didn't get good photos, and it was just a nightmare. So what we did in order to simplify this is we have done it as multiple choice, but what I just do is I say, okay, well, what's the genotypic ratio in this question, or whatever. You still have to do the question, but then you get a specific genotypic ratio, and then that's the answer to the multiple choice question, or whatever. So instead of doing all of the steps. But I do highly recommend, when you are practicing these, when you're doing the practice problems, that you actually write out the entire problem. Now, one thing to mention is that sometimes what happens is that you are given the answer to the problem and you're asked to work backwards. In other words, you may be given a scenario where you're told your let statements, basically, what you're told one is dominant to the other, and these are the two traits. And then they'll say, okay, well, the offspring are 50% this and 50% this. Okay, they'll give you the phenotypic ratios, and then the genotypic ratios are 1 to 2 to 1 or whatever of this. And then they'll say, what are the genotypes of the parents? So in that case, you have to basically do all the steps backwards. You write the phenotypic ratios that you already learned. You'll put them into a Punnett square in the solutions of the Punnett square, and then you have to figure out what the parents would be in order to produce that. Okay, so it's the same process, but you're just running through it backwards to figure out what the parents would be. So I think that in the homework, one of the questions is one way, where you start with the parents and you figure out the outcomes. And then the other one is reversed, where you're given the outcomes and then you have to work backwards to the parents. But it's the exact same process, you just go in reverse. Okay, just for the sake of showing you another example here. By the way, that's a complete question. Right there. That's the whole thing. You've solved the problem. There's the F1 generation and the genotypic and phenotypic ratios of that generation. Just to enhance this and to go a step further, often the questions have a follow-up where they say, well, what are the genotypic and phenotypic ratios of the F2 generation? Okay, so let's say I asked you that. Okay, well, now when you do the second part, you don't need to include the let statements. Okay, so here I've skipped the let statements part. Now we're looking for the second filial, the F2 generation. But I'm looking for, well, I can skip the let statements. We already know what big B and little b mean, and we already used it. So we're going to keep going from there. And now we know, well, we look at the children. These are the children, right? The F2 generation is going to be crossing those children. So the genotypes of the children are big B, little b. We're crossing them to each other, so we already know they're going to be big B, little b by big B, little b. And we know their phenotypes. They're all brown. So this is brown by brown. Okay, and then you do your Punnett square. Woo! Oops, sorry. Okay, and then this is the result of the Punnett square. So we're going to get one big B, two big B, little b's, and one little b, little b. And if we were to look at the actual colors of these, well, that's brown, that's brown, that's brown, and then this one's going to be blue. Again, you don't have to show this on your Punnett square, but I'm just doing it for simplicity's sake here. I can highlight them. And then so when we wrote out our genotypic ratios, this is a 1 to 2 to 1, big B, big B, big B, little b to little b, little b. And the phenotypic ratios, that's step number 6, are 3 brown to 1 blue. Okay, and then again, in your conclusion, your concluding statement, this is a therefore statement. You can write it as a therefore statement. You can say in conclusion, whatever. That's the same thing. Or therefore, 75% of the offspring will have brown eyes and 25 will have blue eyes. That's telling you what the outcome of this mating pair will be. Now, sometimes the question is not just asking what's the genotypic ratio or what's the phenotypic ratio. Sometimes they'll ask a question like, what's the percentage chance of getting brown? In which case, okay, well, you look at this, it's 3 out of 4 times is brown. So 3 over 4, that's 75%, right? So 75% would be brown. Any questions over these? Like how to use this process? It's just the same 7 steps over and over again. So just to go through the steps, what they are, again, there's let statements, write your genotypes, write your phenotypes, do your Punnett square, write your genotypic ratios, write your phenotypic ratios, and then write a let statement, or a therefore statement rather, summarizing the answer to whatever the problem is, whatever they're asking for. And remember that if you're given the end result, the phenotypic ratios and the genotypic ratios, you would just do these in reverse. So you would start with the genotypic and phenotypic ratios, then you'd write out the answers to your Punnett square, and then figure out what the parents must be in order to produce that. And then you're going to work back. So, I mean, you still need let statements, obviously. You still need, sure, I shouldn't say that. You start with the let statements always, but then you're going to be working backwards up to figure out the genotype of the parents and the phenotype of the parents. Okay. Test crosses. What's a test cross? So the purpose of a test cross, and we don't really do this anymore, by the way. Test crosses were originally designed for when you were breeding animals, dogs, horses, whatever, and you did not know if the animal that you had was a purebred. Okay, and this is a problem because if you're breeding purebreds, you need all of the genetic contributors that you're breeding to be purebreds because you want to get the exact same traits every single time you breed them. That's what a purebred is. So if you buy a purebred dog, often you get a certificate of pure breeding. And the certificate is to certify that both parents are certified purebreds. You know that they are homozygous for all of their traits. Now, they don't have to be homozygous dominant, but they do have to be homozygous for all of their traits because, or at least all the traits that you care about for that breed. Not every single trait. I mean, they're not going to be genetically identical, but you want them to be homozygous for every important trait for that particular breed. So do they have the coat that that breed is supposed to have? Is that homozygous, or is that homozygous? Are they homozygous for the size, for the face shape, for the whatever, color, for the temperament, whatever? I don't know. I don't know what they assess dogs for, but obviously this practice is, especially for dogs, really problematic. You'll have an opportunity later if you want to look at dog breeding in more detail. At the end of the unit, there's, not this unit, at the end of the evolution unit, you can look into breeding. We're going to talk about breeding in more detail in evolution. But this practice is a little bit problematic because it dramatically reduces the genetic diversity of animals when you purebreed them. Obviously, you're removing genetic variability intentionally. And if you do that enough, you start to inbreed. Inbreeding is when you are essentially mating animals with their very close relatives in order to maintain these purebred patterns. And when you do that, when you mate animals with their close relatives or other members of the species with low genetic diversity, what happens is a lot of the recessive alleles, these little guys, that are for illnesses, and there are many, all of a sudden start meeting up in the offspring. And you start getting an accumulation of detrimental recessive alleles. These are recessive alleles that normally wouldn't meet in the regular breeding population. It'd be very unlikely for two recessives to come together that would cause a particular illness. However, when you inbreed, your chances of getting two of those recessive alleles together in a baby are much, much higher, which is obviously problematic. So that's why purebred dogs don't live nearly as long as mutts do. Mutts are just random mixes of dogs. So they have a lesser lifespan. They're far less prone to health problems, heart problems, joint issues, pretty much you name it. So purebred animals are much, much more susceptible to a whole host of issues over their lifetime, which is kind of sad, really. But anyway, going beyond that, if you were wanting to purebreed something, and by the way, this works for plants too, so we're not just talking about animals, but if you want to purebreed something, you got to know if you have a purebred. So in order to do that, there is a strategy called test crossing. Okay, we're going to go through this, whoops, a daisy. We're going to go through this strategy right now, this idea of the test cross, and how you actually figure out whether your mystery dog is a homozygote, if it is a purebred. So sometimes you'll see the word purebred substituted for homozygote. So homozygote, especially homozygote dominant, is called a purebred typically, and heterozygotes are also called hybrids. Okay, so there's like a little bit of extra terminology. This is usually only used, this terminology is usually only used when we're talking about breeding for the purposes of breeding animals or plants or something. So a hybrid is a heterozygote, and a purebred is usually the homozygous dominant individual. Okay. If we're not talking about breeding, usually we just use the word homozygous dominant and heterozygote instead of purebred and hybrid. But in the breeding world, in like the dog breeding and the horse breeding world, that terminology is very common, the purebred and hybrid. So just as a heads up. Okay, so the textbook mentions it. It usually uses the term homozygous dominant and heterozygous, which are the actual terms for it in genetics. But for breeders, that's more common terminology. So, but it means the same thing. Okay, so I have two dogs. I'm going to propose a scenario. Okay, one of those dogs, I'm going to call him Buck, is a purebred. Okay, for coat color. Okay, and let's say that for these coat colors, black is the dominant. Okay, they're both black, but one of them is a heterozygote. Okay, Betsy over here is a heterozygote, big B, little b, and Buck is not. Buck is a homozygous dominant, otherwise known as a purebred. Okay, and we want to know which one is which. We're dog breeders. Okay, we don't want to breed a Betsy. Sorry, Betsy, because Betsy is not going to produce a black-coated dog every single time, which is problematic for a dog breeder. Obviously, you want to make the same thing every time. So I got some let statements here. So I'm going to say big B represents black and little b represents blonde. Okay, blonde would be the fur that we do not want in our purebred. And as I mentioned, one of the dogs is a purebred and one of them is not. But which is which? Okay, you can't know that from looking at them. So we're going to do a breeding experiment. And this breeding experiment is called a test cross, and it's for establishing which one is purebred. So what we do is we take both of these dogs and we mate them with a known homozygous recessive. Okay, remember that homozygous recessive here would be, the recessive is little b. Okay, so homozygous recessive would be little b, little b. Okay, all that it says above here is this idea that we're breeding these dogs and we need to ensure that they're purebred in order to sell them. Okay, that's all, basically all that it summarizes at the top there. So in order to do that, we're going to breed each of these dogs with a dog that we know is the wrong thing. Okay, little b, little b, and little b is going to be a blonde dog. Okay, so we breed them both with a blonde dog. Our dogs that we're breeding, we would have to represent in our genetics problem as big B blank. Okay, because we don't know what they are. Now, I mean, I told you what they are, but you don't actually know that until you test. So they're both mystery at this point. They would both be big B blank. Okay, otherwise an unknown genotype. They display the dominant trait, but we don't know that they're homozygous dominant. We hope that they are, because that will make us the money as a breeder. And the other thing that we're crossing it with, in this case, is little b, a blonde dog. Now that's a known dog. We know the genotype of that dog. Okay? So there are two potential scenarios here. Okay? If the dog is a purebred, a big B, and that was the Buck, right? And then the other one over here would be Betsy. Okay, but if we know that it's Buck, okay, Buck is big B. Well, when we breed them, we're going to get a big B and a little b. Okay, that's if we know, that's if it's a big B dog, a purebred. If you do that, and you look at the offspring, every single one of the offspring is going to be big B, little b. Right? You can go through and do the math. So you have a little b, and a big B. Every single one of them is going to be big B, little b. And that means they will all be black. Okay? No matter how many times you breed this blonde one with this black one, you will always get a black dog. So in that case, if you do this and you get a litter of black puppies, every puppy is black, you generally can say that you have a buck. Okay, you have a purebred dog. However, let's look at the other scenario. If your dog is a heterozygote, like this dog here, okay, big B, little b, then what you get is this. And if you look at the phenotypes of this, big B, little b is going to make black, but 50% of the time, you're going to make blonde. So if any of the dogs come out blonde when you breed them, then you know for sure that the dog that you started with is a hybrid. Okay, in other words, not useful for pure breeding. Okay, and this is something that was actually done. This may actually still be done. We can do this through genetic testing now, so you don't actually have to breed the dogs to find out if they're purebred. But traditionally, I mean, that technology has only existed for the past 15, 20 years. So obviously, before that, dogs have been bred for purebred characteristics for hundreds of years, quite possibly thousands of years. So although they didn't do this thousands of years ago because they didn't really know how it worked, but more recently, in the last couple hundred years, test crosses were used to establish if dogs were purebred. This is post-Mendel. So in that way, if any of the dogs that you get show the recessive trait, then you don't have a purebred. Okay, so that's the test cross. You take your dog and you breed it with a known recessive, a known homozygous recessive. If any of the offspring show recessive, then you don't have a purebred. If none of them show recessive, then you probably have a purebred. There's one important caveat here, and that is there are no guarantees in genetics. So Michaela, the question that you're asking is, do you ever get what's called codominance? This is literally the next topic that we're going to address your question, which is what happens when you can get both showing at the same time. Not in this trait, because this trait is completely dominant. It exhibits complete dominance. So you're always going to get one or the other in this particular scenario. But I'm going to present another scenario in a moment where that exact thing that you've just mentioned, a dog with half and half, is actually possible. Okay, but not with this type of trait. So this type of trait that we're looking at right here is called a complete dominance trait, or it's completely dominant. And so when a trait is completely dominant, you can only get one or the other. There is no thing in the middle, a 50-50. Okay, but those traits do exist. It's just not this kind of trait. Okay, so going back to our example here, here's the problem. This is a dice roll. Okay, so when you have a hybrid here, and you breed it with a known homozygous recessive, it's random which one of these you're going to get every time, right? So you roll the dice, you get this one. You roll the dice, you get this one. You roll the dice, you get this one. Then it's this one. Then it's this one. Then it's this one. 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 You'll notice that which one you get every time you roll the dice is completely random, right? In other words, if this was a coin, you'll notice that there's a 50% chance for one and a 50% chance for the other, right? It's half and half. So this is like flipping a coin. It's exactly the same as flipping a coin. So if you flipped a coin 4 times, is it possible to get heads 4 times in a row? And the answer to that question is, of course, yes. Of course you can flip a coin and get 4 times in a row. So it's possible for you to flip the coin and get 4 that are all this. Okay, let's say a dog has puppies and you get 4 and they're all this. Now that's unlikely to flip 4 in a row heads, and in fact, hold on, I can do the math real quick. If any of you have taken finite already, or it's not called finite anymore. There's a senior math course that looks into this exact thing. I have no idea what that course is called anymore in senior math. Maybe you guys know what it is. When you do probabilities, it used to be called finite. Anyway, 0.5, that's a 50% chance to the 4th power. Okay, so that's a 6.3-ish, 6.25% chance. Roughly 6 in 100 times you're going to get this. Okay, if you have 4 puppies. So let's say you get those 4 puppies. Well, that's going to look like this. You're not going to be able to tell the difference between those things. In other words, it's going to look like you have a purebred, but you actually don't. So if you do get all dominant trait, it doesn't guarantee that you have a purebred. It just is highly, highly suggestive that you're looking at this, okay? That this is what's actually happened. But it's not a guarantee. So that's important to remember. There's no guarantee here. However, if you ever see the recessive trait in a test cross, then it's a guarantee. Because the only way to get the recessive trait is if you have a hybrid. So that's important too. If you ever see the recessive trait in a test cross, 100% of the time, you're looking at a hybrid animal that has the dominant and the recessive. Okay, so that's a test cross. That's a way to check to see whether you have a purebred. So what I said here is if all the puppies were black, you can conclude that the dog was probably a purebred. It's almost certainly a purebred. However, there is a small chance that you've just rolled the dice a bunch of times and only got black or only got the dominant trait. Okay, so that is possible, but it's probably a purebred. And the more times you do this, the more likely it is that you're correct, right? Because let's say that you flip the coin, 10 times. You had 10 puppies. If they're all black, the likelihood of getting 10 head flips in a row on a coin, which would be 0.5 to the 10th power, is 0.098%. Okay, so that would be 9. No, not 9 in 1,000. 9 in 10,000. 10 in 10,000 would be, you have a 10 in 10,000th likelihood to get that. That's extremely small. so the more that you get that, the more unlikely it is that and the more likely that it is that you're looking at this. If you get dominant again and again and again and again, it's more and more likely that you're looking at this. Okay, so just again in summary, a test cross is where you cross an individual of a unknown genotype. You don't know if it's a purebred, but it's showing the dominant. Okay, but you want to know if you can breed it so that you cross it with a recessive individual. Okay, one that shows both recessive traits, and then you look at the offspring of that mating, and then you can use that to determine whether or not it is in fact dominant. Homozygous dominant. So when you do these practice problems, and I'm going to get you to do them in a moment, remember that number 1 here is full 6-step strategy, 6/7 steps. 7 would just be the concluding statement at the end, the therefore statement. 1A is you just run through it from beginning to end. 1B is a work backwards, where they give you the answer and you work backwards. And then number 2, again, is a regular use the full 6-step strategy, 6-7 steps, where you go from let statement down to genotypic and phenotypic ratios. So just something to mention here. That's what analysis questions 191, practice 1 and 2 at the bottom of the page. Okay. So what I'm going to do here is I'm going to go through the next component of this, and then I'm going to let you do all the questions at once instead of jumping back, because I think it's more problematic for me to jump back. So I'm going to go through the next part here because it's a perfect answer here to Michaela's question, which is it possible to get the other thing, 50% one thing and 50% the other? In other words, a dog, if it worked like that with this dog, like a dog where half of its fur was black and half of its fur was blonde. Well, not in this case, because this is complete dominance, but there's another type of inheritance, which we're going to talk about right now. Anyway, so what we've been talking about previous to this is the idea of complete dominance. Complete dominance is when one trait is completely dominant over the other, but not every type of inheritance works like that. So what happens if you take 2 solid colored parents, like a black and a white, and then you get a gray kitten a new thing. That happens. So there are traits that are inherited like that as well, that are not complete dominance, where one trait is completely dominant over the other. And you can get a mishmash of the traits put together. Okay, so there are generally two different ways that this can get expressed, okay, when it's not complete dominance. So the first way is called incomplete dominance. That kind of makes sense. So complete dominance is when one is completely dominant over the other. But when one isn't completely dominant over the other, we call that incomplete dominance. It's not totally dominant. And what happens in incomplete dominance is that when you have both traits or both alleles, you get a blend, a little bit of each. And that example that I just gave a second ago is an example of incomplete dominance. In other words, you have a white kitten or a white mom, white cat, and you have a black cat, and you combine them, and when you put them together, you get a gray cat. So gray is not white, and gray is not black. It is a blend of both of them. Gray, a totally new trait, but it is a blend of the other two traits. Okay, so that's incomplete dominance. So in this case, neither of the alleles is dominant to the other. Neither of the alleles is dominant because one allele cannot mask the other allele. And so then this type of interaction in which a hybrid phenotype is made is a blend of homozygous phenotypes, okay, known as incomplete dominance. So the key here is the word blend. It's a blend of the two original traits. So I'm going to give you an example of this happening in nature. The cat example is totally made up, but this example is a real natural example of incomplete dominance. So perhaps you've heard of snapdragons. Snapdragons are a type of flower. I'll show you what it looks like real quick here, so you know what I'm talking about. There's a chapter on this in the textbook that has pictures as well, if you're not sure. They're this type of flower right here. Okay, that's a snapdragon. Pretty. Okay, so the colors of snapdragons tend to exhibit incomplete dominance. Okay, I'm going to talk about two different pigments that are regularly found in snapdragons. Okay, if you're planning to do another example, can you please do number 5 on page 196? Is that one of the homework questions? Is that one of the homework questions, Kim? I don't remember what exactly all of them are. Number 5 on 196. Okay, that looks like it's a it is one of the homework questions for incomplete and complete dominance. Sure, I'll do. Why don't I do that one as one of the examples as part of the note? Okay, I'm gonna jump. I'll jump down and do number 5 together with you. That's totally fine. Okay, so when you are writing out the nomenclature, instead of using capital letters and small letters, what you do in the case of incomplete dominance is you do a base letter. Okay, that would be this one. And that is for the trait. Okay, so in this case, I picked C for color. And then the specific allele is shown as a superscript to that. Okay, so R would be for red in this case. And because it's incomplete dominance, incomplete dominance is shown with only capital letters. So it would be a capital for the trait and a capital letter for the superscript. All capital letters. Yeah. I'll get to it, Kim. I know which question you're talking about, the thalassemia one. Yep. I'm going to get to that in one sec. So I actually think this question will be more clear to you, Kim, once we go over these practice questions here. But it may not be. I'll do number 5 with you. Just give me a moment. Okay. So anyway, they're all uppercase. So for example, if a Snapdragon has red pigment, okay, you could use capital C and then superscript capital R for the nomenclature to show that. Okay, in your let statement, you would say let C superscript capital R be red color pigment. And then for the other one, we're going to look at white pigment here. Let capital C capital W represent the white pigment. And the reason why we use only capitals is because neither of these traits is dominant to the other trait. And so then in the nomenclature, you use only capital letters to represent that they're both dominant at the same time. Okay? There is no recessive one, so they're all capitalized. So this would be an example of my let statements. CR is going to come out red. Okay, and you'll notice that when we're doing let statements for these, we're including what the phenotype looks like for both genotypes. Or sorry, for both alleles. You have to include both alleles, and I'll explain why in a second. So homozygous here, red, gives you red. Homozygous white, that is, they're both white, is going to give you white. That's a white Snapdragon. However, the hybrid, which is both of them together, the heterozygote, is going to be pink. So there's actually 3 different phenotypes that you can get. You can get a dominant phenotype, or actually, now I can't use the word dominant here anymore. You get the red phenotype, you get the white phenotype, and then there is a third phenotype, the pink phenotype, which is the combination of both of these. It's the expression of the white gene and the expression of the red gene, both, or the red allele rather, both being expressed at the same time, and that gives you pink. Okay, so that's an example of incomplete dominance. You've got both dominant traits happening simultaneously, and they produce a brand new thing. That's incomplete dominance. So the reason that you get this is that both alleles are telling the flower to produce, okay, so in CR, will this be test? Are you asking, are there questions about incomplete dominance on the quiz? Yes, there are. I give you some scenarios where you're going to do some practice with incomplete dominance. I'm going to take up some of these questions at the beginning of tomorrow from the homework if people are interested. So if you're trying the practice questions after this and you're like, wow, I do not get this, that's totally fine. I will be happy to review that with you. I have no idea what that means, Trent. So CR, both alleles are both telling the flower to produce red. That makes a red flower. CW, both are telling the flower to produce no pigment. That makes white. But when you have CRCW, you have some alleles telling the flower to produce pink or produce white, and some of telling the plant to produce red. And then the result is that you just basically are producing less red pigment on a white background, which turns into pink. Okay, so CRCW, you end up getting a hybrid with a hybrid phenotype. More importantly is that the phenotype is a hybrid. You get a third phenotype. So if you were to look, remember that if you were to look at this for complete dominance, which is what we were doing before, there would only be 2 phenotypes. There's the dominant phenotype and then there's a recessive phenotype. If there's 3 phenotypes, then you know that something else must be going on. It can't be basic complete dominance if you have 3. Okay, so let's look at, so that is complete dominance. Okay, I'm going to give you another example. Okay, let's say that you had, and by the way, this is not a real trait. The Snapdragon one is good because it's actually real. But let's say you had, let's say that horns on a goat were complete dominant. Or not complete dominant, incompletely dominant. Okay, so that If you had one horn on a goat, sorry, if you had two horns on a goat, two horn. I'm going to write this out up here. So you had a two-horn goat. That's going to be horn 2. If you had no horns, that's 0 horns on your goat, that's going to be horn none. N. Why didn't that right? N. And like I said, generally you indicate the full genotype because you have to have both in order to get the trait. Okay? But if you have the hybrid, that is, you have no horns and you have 2 horns, you get a third completely different thing that is a hybrid of the other two. So in this case, this would be something like 1 horn, or maybe it'd be short horn or something like that. Okay, so you got your 2-horn goat. Oh my goodness, that looks terrible. And you've got your no horn goat. And then the hybrid between the two is maybe a 1-horn goat, sometimes referred to as a unigoat. Oh my goodness, I think my drawing skills are getting worse. There's a little bit of a delay on the iPad right now. It's making it hard to draw, but sure, Trent, ask away. It's just, I'm hoping this is lesson related, right? Trenton, do you feel like that's appropriate to put that in the chat? Oh, wait, is there somebody in the chat using your username? There is somebody in the chat using your username. Sorry. There is another user here who is using the same username as you and posting in this chat, unfortunately. I know. What is it up with people coming into the chat and posting garbage in here? What's the purpose of that? Honestly, like, how pathetic is that to drop in on a science class and just post some garbage in a chat? And people must be really bored. That's just sad. It's all right. It's already, I already kicked that person. I'm trying. Really sad. Really sad. If I could, I wonder if there's some way to whitelist this so that only whitelisted people can join. I'll have to investigate that. That's a real pain, and is a little bit problematic. Sorry to everybody else. Anyway, incomplete dominance is one way where you can get that hybrid expression. There's one other way that you can also get a type of hybrid expression of two alleles at the same time. So that would be codominance. So codominance is when you actually, instead of expressing the two traits and you get a new third trait that's like a hybrid of both, codominance is when you get both of the traits happening at the same time. On its face, this sounds very similar to incomplete dominance, but the actual outcome is quite a bit different. So I'm going to give you an example. Now, one thing to mention here is that when you're writing codominance, rather, you still use an uppercase letter for your base letter, but then your superscript is going to be a lowercase letter. Now you should note that in the textbook, and very annoyingly, in the textbook they don't follow these rules. They don't use a lowercase letter for the superscript, and I believe that is an error in the textbook. They use uppercase letters always for the superscript, whether it's incomplete or codominance, which is very confusing. In every course I've ever taken, in university and in high school, I've always used lowercase letters for codominance in the superscript. So I don't know if that's something that's changed. I would assume it's actually just an error in the textbook, but I'm going to do it this way. But please note when you see it in the text that they don't always follow this rule, which is annoying. Okay, anyway, I'm going to use an example here of hair color in cows. Okay, so there are two different alleles for hair color in cows. There is red hair in cows, which I'm going to show as H superscript little r. Okay, and then there's also a white allele in cows, which I'm going to show as H superscript W, little w. Okay, and there are three ways that this can potentially be expressed in cows. Okay, so you can be a red cow, and this is real, by the way. This is an actual trait in cows. So H little r is a red cow. Okay, that's if you're homozygous red. Or you could be a white cow. H little w is a white cow. Or if you're the hybrid cow, you're something which is called roan. Roan is when you have some white hairs and you have some red hairs. Now you'll notice that this is not the same as incomplete dominance. In incomplete dominance, there would be a third trait that is actually a brand new trait that is some kind of a middle ground here between the two. But a roan cow is actually both at the same time. There's tufts of hair on the cow where some of the hairs in the tuft are red hairs and some of the tufts are white. You can't really see that there. Okay, so it's some white hairs and some red hairs, but both of these phenotypes are being expressed simultaneously. So red phenotype and white phenotype are both being expressed. That's roan. So it's not a hybrid. It's not a new thing that is a combination of the other two. It is both of them being expressed at the same time. So going way back to your question, Michaela, you asked, well, what would happen? Is it possible to get a dog that is showing blonde fur and black fur? Well, if the trait for fur in the dogs is codominant, then yes. Then if you have both alleles, you would get a dog that is some blonde and some black. No. We're talking about, always questions are about interesting edge cases like that. That is actually something different. We don't get into this too much in this course, but I mentioned earlier the idea of being a chimera. So this idea that you have some regions of cells on your body that could potentially have different genes, different DNA than the surrounding tissue. And that's called being a chimera. Sometimes it's a 50-50 direct split down the center. That exists. There's also twin merging, where you have two different fertilized eggs and they merge. And then so there are patches of area, or sometimes a single patch where the genes are actually different than the surrounding tissue. And sometimes it simply happens due to a random error that occurs during the growth of the zygote. So if you can imagine, the cells are dividing. I'm gonna pull up a piece of paper so I can show you what I'm talking about here. So if you can imagine, let's say that you had some scalp here, and the scalp is producing cells that are like, okay, this one's going to make red hair. Okay, these are the scalp cells. And then somewhere randomly, just due to a random mutation during mitosis, while this scalp tissue is growing, you get an anomalous cell that produces white. Okay? This is just due to natural mutation that just happens during this process. Now, that area is, when it does mitosis, is going to expand and produce white, whereas the rest of the tissue that's surrounding it is going to keep doing mitosis and produce red. And so what ends up happening is that you get this patch that produces a different color than the surrounding tissue. And that's just due to a random genetic mutation that happens during mitosis. So that is actually completely separate from what we're talking about, but also produces something similar. That is, it seems like it'd be related, but it's actually not related at all. Hopefully that answered your question, Michaela. Is that like when you have natural highlights in your hair? If you have mostly brown, but then some blonde? I don't know the answer to your question, Caitlin, because hair color in humans is not a Mendelian trait. It is, hair color in humans is there's a number of different genes in your DNA that contribute to your hair color. It's not one single gene. So it is very possible that one or two of the genes that are involved in hair color express codominantly or express incompletely dominantly, in which case you would see some where you're getting a little bit of one or a little bit of the other. A codominant would be the most likely, so that one or two of those genes expresses codominantly and you get this both being expressed at the same time. I don't know that for a fact because I don't actually know the genes that are involved in hair color in humans, and it's not Mendelian. So it's actually a fairly complex process for hair color, unlike tongue rolling and ear attachment and whether other Mendelian traits. I would love to discuss all this stuff as if like, oh yeah, this applies to humans, this applies to humans. But like so much of this does not apply to humans because most of our traits are not Mendelian like this. So it's a little bit difficult to apply this stuff to humans. Kim, I'm going to leave that question for tomorrow because we're going to talk about blood types tomorrow in more detail. So if you still have that question tomorrow, why is blood typing completely dominant? After you do all of the material for the blood type stuff that we're going to do tomorrow morning, then ask that question again, and then I'll go over it with you in more detail. But I do cover it in the lesson, so I'm going to leave it for then. Okay, so why does HR produce red? Well, all the hairs are red because both of the alleles produce red hair. So all the cells are going to be doing one of the alleles, HR. In the HWHW, all of the cells in the coat are going to be producing white hair. But in the HRHW, what happens is some cells are going to express the R and some cells are going to express the W. That's the difference between this and the incomplete dominant. In incomplete dominant, basically both are being expressed at the same time and you get a hybrid. In codominance, the cell is basically choosing. Either it's going to be expressing one or it's going to be expressing the other. And so that cell is going to end up being one or the other. Okay? And so you get both of the actual things showing up on the animal. You either get the W trait or the R trait, in this case, the red or the white. And so some will be red and some will be white. So the expression here is different. So what does this look like in an actual genetics problem? I'm going to, hold on. Okay, I'm going to take this example here, and I'm just going to do it on the next page because it's a little bit too small. So I want you to imagine a scenario where we have a red cow and where we have a white cow. Okay, so I'm going to do some let statements here and say let CR, and notice the small letter R's here. This is for codominance. Is red cow. And CW is white cow. And CW is roan cow. Which again is white and red hairs, both. And what we're going to do is we're going to cross a red cow and a white cow. And I want you to see what that looks like genetically when we cross the two. Okay, so we're going to cross a red and a white cow. That would be the phenotypes of the parents, and I'm also going to write out the genotypes of the parents. So CR and CW. Okay, and then the next thing that you do is you create your Punnett square. So this would be if I was crossing a red cow and a white cow. So we've got CR, CW. We do our Punnett square. CW, CR, CW, CR, CR. And you'll notice, yep, it's 100% CWCR. So then the genotypic ratios here is 100% CWCR, and the phenotypic ratios is 100% roan. Okay, so if you do this experiment, if you breed these cows together, all of the cows that you get from this union are going to be roan. So if you breed a red cow and a white cow together, you get 100% roan. Well, what we can do here is we can look, so this is the F1 generation, okay, or this is otherwise F1 cross. We can also take these offspring and we can breed them, okay? So I'm going to do an F2 cross here and take, or an F1 cross rather, right, an F1 cross, which is taking the offspring of the initial cross, so CWCR, and we're going to breed them with each other. So CWCR and CWCR, which is roan, right? Roan times roan. And what we get then, make our Punnett square. Oh, I want to leave you guys more time to work. I got to go faster. CWCR, CWCR. I'm almost done, by the way. CW, 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 CR, CW, CR, CR, CR. So if you do this in real life, what you end up with is a genotypic ratio of 1 to 2 to 1 of CWCW to CWCR to CRCR, okay? And then the phenotypic ratio is the really weird one, because now I have still 1 to 2 to 1, but it's 1 white to 2 roan to 1 red. Interesting, Kim. Now, I think that if you're finding one single off-color hair, that's far more likely to be caused by a random mutation in that single hair. And interestingly enough, your hair, your head, is being exposed to ultraviolet radiation fairly consistently whenever you're out in the sun, assuming you're not wearing a hat. and it causes mutations exactly like that. So it's not that unusual that a little stray UV ray has knocked some DNA askew and has caused hopefully just a point mutation, a single mutation in that cell, and changed the hair color. That's probably the most innocuous thing that could happen, the least dangerous. Anyway, how do you guys feel about this in general here? The problem solving is exactly the same. We're just changing the way that we're writing the nomenclature for how we list these. And there's two additional ones that you need to know. So there's one for complete dominant. Well, geez, gotta be careful with my language use here. There's one for incomplete dominance. Okay, remember that's capital letters for the subscript and superscript. And then there's, so that's the one where you have 3 traits, but the third one is a hybrid of the other 2. Okay. And then we've also got uppercase base letter and a lowercase superscript for codominance, and that's when both traits get expressed in the hybrid. In other words, a cell basically is going to choose. It's going to do one trait or it's going to do the other trait. Sure, yeah, absolutely. I'll post them as soon as we're done talking here. So one thing I wanted to do, somebody mentioned the thalassemia idea here, which is the idea here is, how do you know what type of expression thalassemia is? Okay, so thalassemia, I better double check this. I don't want to get this wrong. Right. It's when you make an incorrect form of hemoglobin. Okay? So, there are 3 types of thalassemia that you can have. Okay? There's major thalassemia, there's minor thalassemia, or you could be healthy. Okay? So, by the way, this is question number 5 from the analysis questions here from 196, 1 to 3, and 5. I'm just going to do 5 with you real quick. So the question there is, which type of inheritance is this? Well, it can't be complete dominance, because in complete dominance, there is only 2 phenotypes that you can get. You can either have the dominant phenotype or you can have the recessive phenotype, but you can't have anything more than that. It's dominant or recessive. So this is definitely not complete dominance. So then the question becomes, is this codominance or is this incomplete dominance? So we have to think about what's actually happening in thalassemia. So in major thalassemia, what happens is that all of your red blood cells are a weird shape, and they have, well, let's not worry about the shape. They have the wrong hemoglobin, okay? So every red blood cell is producing incorrect hemoglobin. I'm going to draw them as blue red blood cells because they're not actually blue, but they don't hold on to oxygen very well. Okay? In minor thalassemia, some of your red blood cells produce hemoglobin, proper hemoglobin, and some of them do not. And in a healthy person, all of your cells produce hemoglobin, proper hemoglobin, and are healthy. Okay? So, which would this be? Would this be incomplete dominance or codominance? And remember that incomplete dominance is where the middle ground here is a hybrid. In other words, every single cell is producing the same thing, but it's a hybrid of the 2. Or is this codominance, where some cells are producing one and some cells are producing the other? Which one is this? Any guesses? Right. Thanks, Anastasia. This is an example of codominance. So if you were to write this out, if you were to write out the nomenclature for this, and again, you can use whatever letters you want, but I would say I'm going to use B for blood as my subscript, okay? Capitals here. But I would call this B healthy blood. Oh, except it's codominant, so we would use small letters. Sorry, whoop. Better follow my own rules here. Okay, healthy. This would be B thalassaemic. Okay, thalassaemic is when you're not producing the appropriate hemoglobin. And then the middle ground here is where some are producing healthy and some are producing thalassemic. And then that's codominance because some cells are doing one and some cells are doing the other. So as opposed to incomplete dominance where maybe the cells would produce some. Exactly, Lillian. In the case of, if this was an incomplete dominant trait, every single red blood cell would be producing some hemoglobin, and there would be some kind of hybrid thing in the middle where every cell is the same, but they're producing a hybrid between the two, some hemoglobin. But that is not, in fact, what happens. You can use A too. The actual letter does not matter here. Any lettering is fine. So it's because of the way that this is expressed that it would be considered to be codominance. Okay, I'm going to stop talking now and let you get to work. Wow. Again, I feel like I'm more long-winded this quad. That's sorry. You guys are asking great questions though, so it's really not a big deal. So question 5 on page 193 is just a basic problem-solving question, as are number 1 and 2 on page 191. So these are just normal problem-solving questions. This goes in your homework document, by the way. Okay, so those are just some basic practice problems for doing genetic problem solving. Try those, and then I would recommend trying quiz 1 and 2. Okay, do you get it? Are you okay in analyzing the inheritance of a single trait? This is regular complete dominance. Okay, and can you use Punnett squares? Do you know what a test cross is and how to use it? Okay, that's the first part. The second part here is all about using test crosses and incomplete and codominance. There is a really good video on here that goes into more detail in how incomplete and codominance works. And it also, at the end, talks about a little bit about polygenic traits, which we hardly touch in this course, but it's really interesting to look in general at what polygenic traits are and how that works a little bit. So if you're interested in that at all, definitely watch the polygenic traits part at the end. It's not really in this course, so you don't have to watch that part, but I would definitely watch the incomplete and codominance part, which is the beginning of the episode. It talks about how those two traits, how the expression works in those two traits. No, they're all practice quizzes, Julio. So the only quiz that counts is the summative quiz that you do on the last day of the unit. All the other little quizzes that you see posted are just for practice. And the idea is if you're not able to do something or you have questions about something, then you have an opportunity to get extra help if you need it, or you can go back and look at your notes or whatever. Okay, but they're there for you. They're there for you to gauge your own understanding. And then the questions that go along with the complete and incomplete dominance are page 196, numbers 1 to 3 and 5. I just did 5 with you, so you can skip 5. You don't need to do 5. And then there is a practice quiz for those as well for incomplete and co. The last part here, blood types, we're going to do tomorrow morning, so don't worry about it. Okay, and now I'm going to stop talking and let you get to work. All right, if you have any questions, I'll be here.